Good morning and welcome to Samyojana's Spring Symposium. And I'm very delighted that some of our guests are already here. Good evening, Srikanth. I'm very glad to introduce Dr. Srikanth Chalasani, who's a scientist at the Salk Institute in California, in the United States. So sorry for the delay. We did have quite expectedly quite a few technical glitches, and we are dealing with that at the moment. We should start momentarily. So first off, I think I know people are trickling in, but in the interest of time and to ensure that we catch most of the action, I will start off with a little bit of introduction of what this is all about. So Samyojana, I'll give you a little bit of brief history of Samyojana. But again, one more time, welcome to the Spring Symposium today. Samyojana is an undergraduate student-centered initiative. And the main focus has been is the reading, presenting, and discussing primary research literature. And I'll tell you the backstory behind this, this whole effort. The aim of Samyojana is to enable critical thinking and analysis amongst undergraduate students, to instill professional presentation skills and fostering collaboration and teamwork. The story started off when I learned about, through the biology education research literature, a method called CREATE. It was very popular. It was very popular, and it was created by Dr. Sally Hoskins at the City University of New York. It was, while it was not a novel thing to make undergraduate students read, it was not a novel phenomena to make undergraduate students read primary literature. There were a lot of questions about what are effective methods. And CREATE was one of the well-celebrated and published methods that a lot of people started using from about 2005. After that, I had a chance in one of the courses. I was a co-instructor called Science and Technology Honors 199 at the University of Alabama at Birmingham, where I actually saw this in action, where I saw first-year undergraduate students in their first semester get introduced to reading primary research literature. My first exposure to this, when I really first saw this in the beginning, I was actually not convinced that this is the best way to do, where you make students, introduce them to a primary research article and make them read it and discuss. I thought they are not capable of that unless they get a good grounding in fundamental, the core subject knowledge. But to my surprise, I found out that they did extraordinarily well, and that kept happening every year. So with my small interest in replicating some of these experiments in India, we started an experiment in Hyderabad at St. Anne's College in 2019 under the name Integrating Concepts in Biology. And from the students' feedback, we realized it was very, very impactful. And so, as you can imagine, post 2019, we were hit by the pandemic and we had to innovate. So Integrating Concepts in Biology transformed into Samyojana as online meetings, which started off effectively August 2020, I believe, and maybe a couple of months here and there. And very soon it spread from Hyderabad by word of mouth to several places. Right now, I think we can boast about 5 or 6 cities as part of Samyojana, and you will see some of this in action. And along the way, from August 2020 onwards, we will be completing 2 years in this format, in the online format. We tried and tested several formats. And today's symposium is the latest format that we are experimenting with. And thanks to Bhavani, who's here as well from New York University in Abu Dhabi. And again, welcome, Srikanth. So I will now quickly introduce you to the program. So we have this in three sessions. The first one is developmental biology, then we have molecular biology, and then neurobiology. So I would love to get the some inputs and thoughts from both Srikanth and Bhavani in that order. So maybe quickly your initial reflections, and then if you're able to hang out till the end of the first session, we definitely would like to hear more from you, especially after you listen to the presentations. So Srikanth, I know we've been involved in several experiments. So just from the little bit of introduction I gave you, and your own interest in what you see, what you will see a little bit, and maybe initially share a couple of thoughts on what you are expecting, and maybe a couple of your thoughts.
Yeah. Can you guys hear me? Thank you, Anil.
Yep, thanks.
This is a fantastic program. I am really excited to be part of it. I really enjoy talking and meeting with undergrads. I teach also at the University of California in San Diego, and we encourage our first years to start reading primary literature. And I think this is something that I wish I had when I was an undergraduate in India. And I am, to be honest with all of you, a little bit jealous that I didn't have it. So do take advantage of it. And the second thing that I would point out is I am available to be contacted. So please don't hesitate to reach out. I'm a neuroscientist. I study how the brain works in worms and fish and mice, and hopefully in humans soon. So do contact and ask questions.
Thanks a lot, Srikanth. Thank you so much. Thanks. And as Dr. Chalasani mentioned, he's always open to undergraduates contacting him. And I really hope that you will take advantage of that. So Bhavani is a new friend. I met her when I visited NYU Abu Dhabi. And we have been discussing about a few more ideas, and maybe her greater involvement in these kinds of efforts. Bhavani, would you think you'll be able to talk share a couple of your thoughts, or do you want to wait till the end of the first session? It's up to you.
Hi, I'm Bhavani. I'm a research associate at New York University, Abu Dhabi. Sorry, it's a little early here and my kids are just waking up, so I'm not being able to be on video. I am very excited, and I think it's a great opportunity to start critical thinking as early as possible. And I second Srikanth that it's I didn't get to do this when I was younger, and I think as the earlier we start, the better it is, because you gain much more confidence, and then you can read the complicated papers over time. You start with the simpler one and slowly build up over time. I am just excited, and I want to listen to a few talks, and then we can discuss.
Thank you, Bhavani. So without any further delay, we are just about 3 minutes behind our schedule. So Varni, Anahita, and Sahil, please take it away.
Good morning, everyone. Is our screen visible?
Yes, it is.
A very good morning to all our esteemed guests and peers. I am Sahil, and I, along with my teammates Vani and Anahita, are going to present a paper in a very interesting field, which is of maternal-fetal relations, focused on the imprinted IGF2-IGF2R axis being extremely critical for matching placental microvasculature expansion to the growth of the fetus.
Sahil, I'm just sorry to interrupt you right at the first slide. I just want to let everybody know Sahil, Anahita, and Vani are first year undergraduate students at the Shiv Nadar University doing their BSc in biotechnology. Sahil, back to you.
All right, so let's talk about one of the main characters of this story, the placenta. So it's basically an organ that develops during pregnancy and is responsible for meeting the basic demands of the fetus like nutrients, hormones, etc. And the placenta is composed of both maternal tissues, which are collectively called the decidua, as well as fetus-derived tissues called the chorion. Now, let's take a look at the main focus points of this paper. So it's to identify certain fetal signals responsible for placental development, to understand the genes responsible for creating this system of signaling, and to correlate these signals with the increasing nutritional demands of the fetus as pregnancy progresses.
The main genes focused on are IGF2 and IGF2R. The IGF2 gene provides instructions for making a protein called insulin-like growth factor 2. This protein plays an essential role in cell survival, metabolism, proliferation, and differentiation during the course of pregnancy. So how does the IGF2 protein exert its effects? It binds to certain receptors like the ones listed below. The receptor it binds to with highest affinity is IGF2R. IGF2R has the ability to bind to both IGF2 and mannose-6-phosphate. Once IGF2 binds to IGF2R, there can be two possible outcomes. It could lead to a system of signaling by which it exerts its effects, or the receptor could carry the IGF2 protein to a lysosome for degradation. One of the important concepts that is crucial in understanding this paper is genomic imprinting. Genomic imprinting is an epigenetic phenomena that causes genes to be expressed or not expressed depending on whether they are inherited from the mother or the father. So how does imprinting work in the case of IGF2 and IGF2R? In mice, IGF2 is maternally imprinted, which basically means that the maternal allele is silenced, and so the IGF2 protein produced is due to the expression of paternal allele. Contrary to this, in mice, IGF2R is paternally imprinted, which means that the paternal allele is silenced, and so the IGF2R protein is due to the expression of the maternal allele. In humans, however, IGF2R imprinting is a polymorphic trait, and so there have only been a few cases identified where maternal expression and paternal silencing have been seen. Two syndromes associated with IGF2 genomic imprinting are Silver-Russell syndrome or SRS, which is caused due to a reduction in IGF2 gene expression, and Beckwith-Wiedemann syndrome, BWS, that is due to the loss of maternal imprinting and biallelic expression of IGF2. The patients with BWS showed somatic overgrowth and were more prone to tumor formation.
Coming to early embryonic development, allow me to talk about the physiology of early embryonic development. Upon reaching the blastocyst stage, as we can see, the inner cell mass differentiates into two different clades of cells, one being the primitive endoderm and the other being the epiblast. The epiblast clade of cells gives rise to three germ layers of the mammalian body, which are the ectoderm, endoderm, as well as the mesoderm. The mesoderm then differentiates to form different organs as well as the endothelial cells. Now, let's discuss some terminology that we might come across in this paper. Starting off with labyrinthine zone, it is an embryonic derived structure in the placenta. It is basically an interface for nutritional and gaseous exchange. It increases with size with advancing gestational age. Coming to what the labyrinthine trophoblast is, it is an extra embryonic component of the labyrinthine zone itself. Then we come to the maternal blood spaces. It is an intervillous space of placenta which is filled with maternal blood. It is formed by the rupture of maternal capillaries, an event that is in fact stimulated by the fetus itself. A word, angiogenesis, is something we will come across multiple times during this paper. The formation of new blood vessels, which involves migration, growth, and differentiation of endothelial cells, is what the term angiogenesis refers to.
So, how can the effects of IGF2 and IGF2R be studied? By creating mutants. So, the group of researchers performed mutations in different types of lineages of cells. Let's consider the first type of mutant. As indicated in blue, these mutants have been created by knocking out the paternal IGF2 allele from the epiblast lineages. And as previously discussed, the epiblast is the clade of cells that gives rise to all bodily tissues. It, along with fetoplacental endothelial cells, are made devoid of IGF2 and are hence named the IGF2 epicomutants. The second type of mutants studied were the IGF2 ecomutants, which were created by knockout of paternal IGF2 allele from the endothelial cells and the fetoplacental endothelial cells. So this would mean that since the maternal allele is already silenced, there's no IGF2 being produced in these cells. Similarly, 3 other types of mutants were produced by knocking out the paternal IGF2 allele from the trophoblastic cells and hematopoietic cells. The IGF2 TRKO and IGF2 HCKO mutants were created. Finally, they also knocked out the IGF2 ubiquitously from all cells to produce the IGF2 UBKO mutants. The effects observed by knocking out IGF2 from epiblast lineages were placental growth restriction from the 14th day of gestation, compromised LZ expansion between the 14th and 19th day, abnormalities in the volume, surface area, and length of fetal capillaries from the 16th day, reduced FPEC count from the 16th to the 19th day of gestation, as well as a reduction in the volume of labyrinthine trophoblast and maternal blood spaces. Further, they noticed that certain genes were responsible for controlling immune responses and impairing cell proliferation, as well as angiogenesis, were upregulated. In the graphical representation, we can see that, as indicated in blue, the IGF2 epicomutants are seen to have a significantly reduced rate of fetal growth, as measured by their weights when compared to the controls. A similar effect was seen in the placenta. A growth restriction measured by the placental weights was observed on comparing the mutants to the wild-type individuals. The effects observed by knocking out IGF2 from endothelial cells, although not as severe, was similar to the ones seen in the IGF2 epicomutants. Placental growth restriction reduced labyrinthine zone and labyrinthine trophoblast, reduction in the maternal blood spaces, and fetal capillary volumes on the 19th day of gestation were some of the effects observed. Once again, the genes responsible for controlling immune responses and impairing cell proliferation, as well as angiogenesis, were upregulated. We can once again see a reduction in the rate of fetal and placental growth measured by their weights in the mutants, as indicated in blue, on comparison with the controls. However, as shown by the graphs, these reductions are not as significant as the ones seen in the IGF2 epicomutants.
Certain inferences were drawn from other mutants that were studied. On studying the IGF2 HCKO, PRKO, and the UBKO mutants, no significant results were found. The hematopoietic-derived IGF2 was seen to not be required for the expansion of the labyrinthine zone during late gestation at all. As well as the TRKO mutants, the FPEC expansion is not seen to be dependent of the trophoblast-derived IGF2, thus making us conclude of its irrelevance. Coming to the UBKO mutants, the trophoblast-derived IGF2 did not contribute significantly to the FPEC expansion.
Now, let's see how IGF2R, that's the receptor, fits into this picture. To understand this, two experiments were conducted, one on the fetal cells and the other on the fetoplacental endothelial cells. So, as previously stated, we know that IGF2R is produced due to the expression of the maternal allele and that the paternal allele is silenced. So, coming to the first experiment, the maternal IGF2R allele was knocked out from the fetoplacental endothelial cells. So they observed a reduction in the proliferation of the FPECs in the presence and the absence of IGF2. So they inferred from this that IGF2R is actually required for the normal proliferation of the FPECs with no direct correlation with the presence of IGF2. Next, they performed experiments to find a correlation between this IGF2R signaling and a pathway that's actually commonly involved in cell communication, known as the ERK pathway. So through its signaling mechanism, this pathway produces some changes in the cell, like cell division. And so how is the IGF2R signaling related to this ERK pathway? So to understand this, whether cell proliferation could be controlled by the ERK pathway, they knocked down the genes involved in ERK signaling. So they observed that there was an upregulation of certain genes that were also upregulated in the mutants produced by knocking out the maternal IGF2R allele. For example, a gene that's involved in causing anti-angiogenic effects. So seeing this, they then inferred that the IGF2-IGF2R signaling process promotes FPEC proliferation through the ERK pathway. Now, coming to the second experiment they conducted, the maternal IGF2R allele was knocked out from the fetal cells this time. So they observed an increase in extracellular IGF2 and body overgrowth as well, and perinatal lethality. So they inferred from this that IGF2R mediates the circulating levels of IGF2. So to explain this, let's actually revisit those two possible outcomes once IGF2 binds to its receptor IGF2R. So as we can see here on binding, this could lead to a system of signaling, or the receptor could carry the IGF2 protein to a lysosome for degradation. Therefore, in the absence of IGF2R, which is the mediator of IGF2, as we have found, there is an increase in the growth of fetal cells, which could happen because IGF2 exerts its effects through other receptors. We know that it binds to a bunch of other receptors and without getting degraded in the lysosome. This is actually, in fact, similar to the Beckwith-Wiedemann syndrome that we talked about, in which overgrowth was seen due to a biallelic expression of IGF2. So we can now summarize these two experiments and the effects of IGF2R studied. So in the fetal cells, they noticed that the deletion of the IGF2R maternal allele caused increased cell proliferation and overgrowth due to the lack of lysosomal degradation. And in the FPECs, the fetoplacental endothelial cells, on the other hand, the maternal IGF2R allele deletion caused reduced proliferation. So based on their correlation of IGF2R signaling and the ERK pathway, we think that IGF2R primarily kickstarts these cascade of proteins that are involved in ERK signaling, thus increasing cell proliferative activity, which is why its deletion in the mutants led to a reduction in this proliferation.
Based on their findings, a model explaining the effects of the circulating levels of IGF-2 was proposed. The relationship between the increasing fetal demands, placental supply capacity, and increasing levels of fetus-derived IGF-2 turned out to be not just a correlation, but also a causation. The effects of circulating IGF-2 were identified to be endothelial cell proliferation, capillary branching, trophoblast growth and differentiation, and fetal capillary growth and remodeling using the reservoir of IGF-2 in an autocrine and paracrine manner. Now let me introduce you to the words autocrine and paracrine. Autocrine signaling is when a cell targets itself, releasing a signal that can bind to receptors on its own surface. Paracrine signaling occurs when a cell targets a nearby cell. Now, coming back to the model, they suggested that fetoplacental endothelium is a large reservoir of IGF-2, boosting further IGF-2 signaling and acting in a paracrine and autocrine manner, as I talked about, controlling the growth and remodeling of the fetal capillaries. As can be seen in the figure, IGF-2 binds to the endothelial cells to stimulate the production of certain signals that further cause more IGF-2 production in an autocrine manner. The endothelial IGF-2 also affects trophoblast cells in a paracrine manner to release more IGF-2. Importantly, the effects of IGF-2 signaling on fetoplacental microvasculature remodeling seems specifically driven by fetus-derived IGF-2, as they did not find any evidence that the IGF-2 provided by the trophoblast has any direct role in vascularization. They therefore suggested that the key role of circulating IGF-2 is to provide fetus-derived angiogenic signals in order to promote the vasculatory expansion during later stages of the gestational period. To conclude, we can say that the imprinted IGF-2-IGF-2R axis is extremely crucial for matching the placental microvasculature expansion to the growth of the fetus. Coming to a few questions that were left unanswered. Firstly, they spoke about the effects of the upregulated genes seen in the mutants, but from their graphs, we can see that there are other genes that have been downregulated. So how exactly do these downregulated genes affect the growth of the microvasculature development and the fetoplacental endothelial cell development in the mutants is something we are unaware of and is left unexplained. Secondly, on deletion of the IGF-2R maternal allele from the endothelial cells, a reduction in the total ratio of the fetoplacental endothelial cells to the area of the labyrinthine zone count was seen at the 16th day, but became normal at the 19th day. And what possibly could explain this anomaly? We personally believe that this normalization of the FPEC on the 19th day of gestation could be possible due to an increase in the functionality of other receptors that are receptive of IGF-2, thus allowing IGF-2 to exert its effects. With this, we come to the end of our presentation of this paper. Coming to what we enjoyed the most about this paper, I personally developed a new interest in the field of developmental biology, which I would like to explore more in the near future, and that's for sure.
I think the part which I loved the most in the whole paper was when we had to understand the concept of genomic imprinting, and I would definitely like to explore the area more.
And I think what I enjoyed learning the most was about how these different signaling mechanisms control the levels of circulating IGF-2 and how they just work as one unit to sustain such a healthy fetus. And as a group, what I think we enjoyed the most about this paper was to learn about exactly how a change in one small aspect of an entire system can have such wide implications on its normal functioning. And we further thoroughly enjoyed working as a team to break down the story being told by this paper and just helping each other understand all the finer details.
And I'd also like to talk a little bit about the challenges that we faced along the way. we found it quite hard to understand certain topics in the paper. For example, genomic imprinting. This is a topic that we had never been introduced to before and also happened to be one of the key concepts in this paper. Another topic that we encountered for the very first time was the ERK pathway. And we realized how this was such a major system of signaling that's involved in so many different processes. This, as well as understanding the entire picture of embryology as a whole, was some of the conceptual challenges we encountered as a team. I think by reading extra information on these topics and more papers that we read helped ourselves understand them better. There were certain aspects that weren't talked about too much in the paper, which we couldn't understand to a very deep level. I think we feel that if we learned the basics of these aspects and got assistance from Sir by spending time to really try and understand it.
And I feel that from now, we will actually come across different kinds of research papers, which we would probably have to deal with using different strategies. So one challenge that we'll probably carry with us on our journey now would be to keep working out new methods to approach papers and topics in science in general in a way such that we always maintain our interest in learning new things, however complex they may be.
So before officially ending our presentation, we would like to thank Dr. Ranil Challa for bringing Samyojana to us and really helping us learn so much in just a few months. So we thank you for this entire journey, sir. And we would also like to thank all our fellow Samyojana peers. We're glad we were introduced to students from different institutes to work together on such a wonderful project. And so we hope that we sparked an interest towards maternal-fetal relations in all of you. Thank you. Now we'll open the floor for questions.
Thank you, Vani, Anahita, and Sahil. As you said, the floor is open for discussion. So I know Dr. Srikanth has to leave. It's quite late in California. So Srikanth, do you want to have a couple of questions and maybe give some suggestions? Yeah.
So that was really good, Vani, Anahita, and Sahil. Sorry, I mispronounced your name. So I have a couple of general questions. Who assigned this paper to you? How do you pick this paper?
So I think when we saw the list of papers which were available to us, we just saw the name of the topic of the paper, and we had talked to one of our professors initially, our animal sciences professor, and she told us about the conflict between the maternal, the mother and the fetus. Mm-hmm. So I think that was one part that
it really helped us. We immediately were attracted to this paper specifically and wanted to work on it.
Okay. So can I ask the three of you, what is the central hypothesis that the paper is trying to test?
I think the paper is trying to understand exactly what these signaling mechanisms are. They probably already know these genes and that they have some sort of role in helping expand the placenta and helping the fetus grow. But the researchers wanted to delve deeper into how it was actually being carried out.
And I think they specifically wanted to focus more on IGF2. I think they might have had some sense of idea that maybe IGF2 has a more important role than we believed earlier, and they wanted to focus on that.
Also, probably they would have wanted to convey the importance of the fact that it is the fetus-derived IGF2, because IGF2 is something that is provided by the maternal blood as well. And in fact, in a different paper that we were reading during the course of Samyojana, they did seem to have an effect on the development of trophoblastic cells, but then the placental microvasculature as a whole being dependent on IGF2, it is something they wanted to show that it is the fetus-derived IGF2 and the signaling that is stimulated by the fetus that proves its importance.
Okay. So would you agree that as the fetus is developing, it's going to need additional resources?
Absolutely, sir. As the
Okay.
fetus keeps developing, it needs more and more nutrition, more and more gaseous exchange.
Okay. Now, is it possible that the receptor that senses the fetus derived signal is located in different different compartments of the placenta?
So could you
So,
just
you
repeat your
presented an experiment. You showed us 2 experiments where you said the IGF-2 receptor is either in the endothelial cells or it's in another cell population. The endothelial cells had an effect, but the other cell population knockout did not have an effect. Isn't that true? Is that true?
Yes, sir, that's absolutely true. In fact, the effects of the that were seen in the placenta, they were effects that were significantly seen in those compartments of the placenta that were derived by the fetus. The fetus-derived cells that were in the placenta showed all these significant effects. The maternal-derived places, of course, are affected, but to a lesser extent, because after all, even they form by stimulation by the fetus, but then the significant effects were those of the fetal side of the placenta.
So the way I would recommend, I mean, obviously you can read a paper in any way you like. The way I would recommend this is to look at each experiment and ask, what was the central hypothesis? So the first experiment that you talked about was a knockout of the receptor in the endothelial cells. So the hypothesis of that experiment is that the endothelial cells are sensing the ligand that's being released by the fetus and resulting in more blood flow or new blood vessels to develop. And if that was your hypothesis, what was the experiment that you designed to test it? The experiment you designed was to see if you took the receptor and you deleted the receptor only in those cells without affecting the receptor anywhere else, what would happen? And the observation you got was that when you deleted it specifically in the endothelial cells, you affected the biology that you were studying. So when you start to think about each part of your paper as a hypothesis or a question, the design of the experiment, the result that you got, and then you can ask, did that result explain or give you more hints on what that question was that you began with? So that's how technically how all of these papers are being written, or at least how I would recommend for you to read it. So I have two other comments I want to make. One is imprinting. Imprinting is a super cool phenomenon, right? In the context of genomics, in the context of development, imprinting is basically the idea that you inherit a gene that is expressed either maternally or paternally. And the second comment I would make is that IGF-2 is a really fascinating gene because it's one of the few genes that are paternally inherited in humans and in mice. We don't know why, but that's something maybe you guys can figure out for us. So I'm glad you're interested in developmental biology. Developmental biology is a beautiful part of science. The idea that a single cell can develop into a complex organ, into a complex animal, with all of these tissues is something that we all aim to understand. That makes sense to you?
Sir. Yes, sir, definitely. And something that I think got us hooked to developmental biology, it's something that we came across in our previous semester, in fact, when we were learning a bit about paternal and maternal imprinting, just a bit. We came to know about certain facts, like for a male organism to want to transfer its genes to its offspring, it will have to have a certain amount of selfishness in its genes, and therefore genes that the paternal the father gives to its offspring, they will be such that they will like cause the fetus to invade the maternal tissue and absorb as much of nutrition possible. And the fact that there's an element of selfishness that promotes this relation that we see as so intimate, it was definitely something that got us hooked to this topic. And I think it applies for IGF2 as well. The fact that it's paternally imprinted really uh, sparked that idea in me that it's probably because the father would want his gene to be carried out forward. So it will have to do something so that the fetus does not die. And IGF2 is something that stimulates the microvasculature development, invading the maternal tissue and like helping the fetus grow, like ensuring its survival, basically. All of these connections that we were making while reading was something that really intrigued us.
Also, just finding out for the first time that like you would expect the mother to want her fetus to be very healthy, but finding out that she would prefer to distribute her resources while the father would want each fetus to be very healthy. So there's this kind of conflict between the mother and the fetus was very interesting, which is something that attracted us towards this paper very much.
Yeah, so I want to make one more comment. I'm sorry, my last comment. So I would urge caution when you try to make these broad connections and correlations. So always look at what the data is really telling you. All your data is telling you is that knocking out this receptor in one cell type or one cell population has an effect. In the second cell population, it doesn't have an effect. Now, the authors of the paper are using that data to propose a hypothetical model. The hypothetical model is what you're talking about. That model does not have data right now. Now, maybe there is a follow-up study that might have data for that. And that's true of most papers. Most papers will take an observation, take their data, and try to place it in the larger context of what it would mean for the community and for the field. But that larger context hasn't been proven yet. So as a scientist, and you're all scientists now, as a scientist, you look at your data and you can only conclude what the data is telling you to be true. And even then, if there is data that says that it's not, then obviously your hypothesis changes. Okay?
Yes, sir.
Okay, good luck, guys. This is really fun. I'll try and
Thank you
log
so
back
much.
in later, but if I don't log in later and you do want to reach out, ask Anil. He has my contact information, and I'm happy to communicate with all of you.
Thanks, Srikanth. Thanks a lot. Wonderful.
Bye.
Bye. And we do have a few minutes for maybe one more question from the audience. Question or comment? I do recognize a few guests coming in. So if people would like to chime in, and if you don't have any questions, you can give your suggestions. Like Dr. Srikanth has mentioned, any observations or suggestions or advice that you have for the students.
Hi, Anil.
How
This is
please
Bhav-
go ahead.
Yeah,
Yes.
I really like Bani and Nahita and Sahil. Very good attempt. Very, very nice. I'm impressed that, the youngsters can do such good job with the PowerPoint presentations. One thing is like you put the sources for the images right now, you have to put the citation in every slide as you go through the introduction. So, you have, where to get it from. Also, I found that, when we discuss a research paper, it's usually have to put the figure. If you put the figures from the paper in the presentation and explain that with less text, it makes more of an impact because the visual representation is a lot more, and it is the data that is presented in the paper, right? So if you can put the figures and then a little bit of less text would help a lot more. Also, when you start out the presentation, this is just technical stuff. These are when you start with the idea, and then you slowly build the idea like a flow chart. So, and then you can go back to the flow chart, like step 1, IGF, then IGF receptor, and then you talk about the various layers of the tissue and the maternal and the fetal effect, and then you keep building the flow chart. So in the end, we have an idea of how the paper flows. And at the end, you end like Shrikant said, with the hypothesis, because nothing is like finalized in research. We just come up with more questions at the end, and we come up with another hypothesis to test further. So that would actually help with with making it even more clearer to understand the overall impact of the paper. But you did a really good job. These are just a little more suggestions to make it even better. Thank you.
Thank you,
Thank you
Thank
so
you,
ma'am.
much.
everyone.
You, ma'am.
Thank you very much. I think we are on time. So let's go to the next presentation by Adrija, Akanksha, and Sasya. And please let the audience know your affiliation and which, what you're studying. This is some kind of a self-promotion for Samyojana about the diversity of places that you're all coming from.
Good morning, everyone. My name is Akanksha Bhattar. I'm an undergrad student at Banaras Hindu University. Today, I, along with my teammates and classmates, Audrija Goswami and Shasya, will be presenting a paper titled Limb Development Gene Underlie Variation in Human Fingerprint Patterns. I'll start by giving a brief introduction on fingerprints. Fingerprints are a set of ridges and lines present on the skin of palm and fingers of the hands, as well as on the soles and toes of the feet. So this
Is somebody checking the slides? We still are on the first slide.
Yes, sir. So this is the first slide.
Okay, fine. As long as people are paying attention to the slides, you're good. Go ahead.
So this features uniqueness, that is, fingerprints are unique to every individual and it is not repeated. This makes it intriguing and a research topic. Also, Dr. Chandana Basu ma'am, who is one of the co-authors of this paper, is from our very own university. So this was also one of the reasons which made the three of us choose this paper. Now let's start. By discussing some basics of fingerprints. So let's start by the type of fingerprints. So there are three principal types of fingerprint patterns, which are arc, loop, and whirl. Although fingerprint patterns probably evolved to help in grasping and sensing surface tension, but since 19th century, they are widely used for personal identification because of their uniqueness in each individual. So the ordinal phenotype for arc is 0, for loop it's 1, and for whirl it's 2. So the basis on which these patterns, these fingerprinting patterns are classified is the number of triradii, which is indicated as a blue triangle, and the number of cores, which is indicated as the red circle. So depending on their numbers, they are classified as arc, loop, and whirl. So each main group has two subtypes. The arc may be simple or tented. The loop may be ulnar or radial. The whirl is either simple or double. So now let's try to understand some basics of limb development process briefly before we go on to the experiments and the main part. So these are the ECGs of different developmental stages. So the figure A looks like a paddle with protrusions as seen. Now these protrusions are the future fingers. So in the figure B, which is almost 6 to 8 weeks, it is seen that the fingers have grown apart. So so these in the figure B, we can see some outgrowths at the distal ends of the fingers. So these outgrowths are called the volar pads. Now the volar pads are formed in the 6th week and it's get completely formed in the 12th week. So here is a illustration of how the volar pads are formed. So as it can be seen that it starts forming in the 6th week, and the formation is complete in the 12th week. So after its formation, the regression of these volar pad start in the 13th week, and the regression takes place till the 17th week. So it is these regressions which are responsible for the ridge formation. Although there are many mechanical theories which are which has been proposed for the ridge formation. For example, the resolution of mechanical stress on the epidermis through buckling. Now, this is a cross section of dermis. So the first layer is epidermis. The second one we have below epidermis is the basal layer, and the third one is dermis. So the basal layer, the cells in the basal layer start growing at a rapid rate. They start dividing rapidly, and due to this division, there is a stress which is formed in these basal layers. Now, this stress causes buckling, and as a result of which ridges are formed in the dermis layer. And we have many mechanical theories which are given for the fingerprint, for the variation in fingerprint development, but we don't have any biological explanation for this. So this paper basically tries to explain the biological reason behind these fingerprinting, behind the differences in fingerprints.
Since genome-wide association scans are used in a wide range in this paper, so before going any forward with the presentation, it's good to discuss a little about genome-wide association. So genome-wide association is an approach that helps scientists to identify genes associated with a particular disease. They study the entire set of DNA of a large group of nucleotide polymorphism, which is SNP. So this is the graph of a genome-wide association scan, and it is known as Manhattan plot. So this is interesting. So does anybody knows why it is called Manhattan plot, or want to make a guess? Anyone? So this is called Manhattan plot because this graph resembles the city line of Manhattan, city of Manhattan, skyline of Manhattan city. As there skyscrapers towers of the lower and variable height buildings. So do Google, guys, it does resemble the Manhattan city skyline. So in this graph, x-axis represents the haplotypes from each region of the genome that are tested and organized by chromosomes shown in different colors by blocks. So haplotypes are the combination of allele or a set of SNP found on the same chromosomes. Here, this graph represents thousands of dots, and each dot represents one haplotype. Here, in this graph, y-axis represents the relative frequency over the association of haplotypes in that region of the affected individual compared to the control individual. So higher the value of P, the stronger is the association with the trait. So researchers conducted genome-wide association scans on fingerprint pattern of all 10 digits in Han Chinese cohorts. These cohorts included three study groups, Taizhou longitudinal study, National Survey of Physical Traits, and Zhejiang cohort study, and it included 9,909 individuals. So GW is a scan on ordinal phenotype, which is Arch, Loop, and World. They identified 18 genome-wide scans, 18 genome-wide scan signals, and other studies show the similar results, and majority of signals, which is like 17 of 18, were replicated in other. This Manhattan plot, x-axis represents chromosome and y-axis represents frequency. And here they have shown 18 genome-wide signals along with their association significance across different regions. So here each block represents each digit. Gray box shows the marginal relation of that gene with a particular digit, and whereas the black represents the significant association with a specific digit. They further conducted GWS on different phenotypes, and results overlap the 18 before mentioned signals. It suggested that ordinal phenotype covers the most phenotypic information related to variation in fingerprints.
Can you be closer to the microphone? Your audio is quite muffled. So if it can be made better.
Is it better now, sir?
Slightly better, yeah. If you can get closer to the microphone, if possible.
Now?
One more time.
Is it okay now?
It's okay. Yeah, continue. It's fine.
Okay. So the fingerprint pattern types on the left and the right edge of pairs are highly correlated. They also have a strong genetic correlation. So in this slide, we talk about pattern blocks. What is pattern block? Pattern blocks on the middle three digits, patterns on the middle three digits are more correlated to each other than on the patterns, then are the patterns on the rest of the digits, which shows a pattern block or pattern influence phenomena. With the help of previously discovered 18 GWS genome-wide scans, researchers found that genetic correlation among the middle three digits is significantly higher than among the other digits. While this can easily be explained by this graph presented. In this graph, y-axis represents the correlation coefficient. RT means phenotypic correlation coefficient, and RG means genetic correlation coefficient. So here blue color represents phenotypic correlation and red represents genotypic correlation, and it can easily be comprehended that the pattern block pairs show higher correlation than the other pairs. Here it is again a jargon, topologically associating protein. It is a self-interacting genomic region. In this figure, we have shown a nucleus with chromosomal territories. Chromosomal territory is basically a location where a particular chromosome resides more frequently than other places. In figure it has zoomed in a particular chromosomal territory and have shown 5 tags. 2 of them are repressed and 3 of them are active. So the hexagonal structure between tags are the tag boundary. A lot of crosstalk is found in tag. DNA sequence within a tag physically interact with each other more frequently than the sequence outside of the tag. The crosstalk like the correlation between promoter and enhancer is usually seen in the tag. With further research, they found that the locus EBI1 contributes in the pattern block. The top signal of this locus is found 100 kilobase downstream of the EBI1 gene, or also known as NECOM, and discovered 2 SNPs, RS764687 and RS763083. And they also found that the SNP RS764897 and EBI1 gene share are in the same tag. As I've already explained, that the tag genomic sequence within a tag interact more than the genomic sequence outside the tag. So the lot of interaction has been found between EBI1 and this SNP. And actually the interaction between the promoter region of EBI1 gene and SNP is seen. As in this graph, we can see that it shows the long range of interaction between genomic regions and chromatin interaction between the promoter region of EBI1 gene. Hence, it shows that the SNP RS764897 helps in the modulation of expression of gene EPI.
Okay, so on the fingertips, the regular patterns of ridges and furrows form fingerprints. So what causes ridges to form? This is figure A, which shows dermal surface of palm stained with toluidine blue from wild type and mutant EVI1 adult mice showing dermatoglyph arrangement. Here the arrow indicates spur on the mutant digit 5. Before proceeding further, I want to tell what is dermatoglyphs. The parallel ridge formations present on the skin of the palms and fingers of the hands and on the soles and toes of the feet are called dermatoglyphs. And EVI1 plays a major role in dermatoglyph patterning. Figure B is a schematic depicting transverse ridge categories on mouse digits and ventral surface of digit 4 of wild type and mutant EVI1. Here the C region indicates continuous ridges, D indicates discontinuous ridges, and I indicates incomplete ridges. Next slide. This is a graphical representation showing quantification of digit ridge pattern in wild type and EVI1 mutants. Here x-axis represents ridge count and y-axis represents ridge categories on digit 2, digit 3 and digit 4. The blue bar indicates wild type and red bar indicates EVI1 mutant. Here it was found reduced frequency of continuous ridges in all mutant digits, with digit 3 and digit 4 also carrying more discontinuous ridges. So this results demonstrate directly that EVI1 itself is a modulator of dermatoglyph patterns. Next slide. EVI1 mutation alters mouse dermatoglyph patterns and is expressed through early limb development stages. It was assessed EVI1 expression in intact embryonic mouse limbs and identified that EVI1 is broadly expressed in autopod at embryonic day 12.5, that is E12.5, and then becoming restricted to the distal regions of all emerging digits. Broad EVI1 expression was also identified throughout limb mesenchyme at embryonic day 11.5, then becoming restricted to the distal end of the limb at embryonic day 13.5 and excluded from the cartilage elements of the digits by embryonic day 15.5 and reducing further by embryonic day 17.5. Next slide. This is a graphical representation showing EVI expression in mouse forelimb at embryonic day 11.5, 13.5, 15.5 and 17.5. Here we find a steep decline across the stages of limb bud outgrowth at embryonic day 11.5, digit emergence at embryonic day 13.5, digit outgrowth and definition of dermatoglyph patterns at embryonic day 15.5 and 17.5 respectively. Next slide. The transverse digital regions in the mouse was analyzed as the closest model phenotype of human dermatoglyphs. EVI1 expression was also determined during human embryonic development by immunofluorescence. Figure G shows the transverse section of C17 embryo, that is 6-week estimated gestation age. This shows broad expression in the mesenchyme of the early outgrowing limb bird at this stage. And here EVI1 expression is represented by pink color and DAPI stain. In figure 8, this is a longitudinal section of 10-week estimated gestation age digit. Here the arrow indicates raised volar pad across which fingerprints form. By 10 weeks, EVI1 was prominently expressed in the mesenchyme of the distal end of the digits, notably under the volar pads, and this can be clearly seen in this figure. Next slide. This is a 13-week estimated gestation age digit where expression is largely lost, being present only at the periphery of the distal phalanx. Here you can see K14, which is an epithelial marker and is represented by light green color. Next slide. This is a 16-week EGA digit detecting EVI1. At 16 weeks, as dermatoglyphs are emerging as a periodically corrugated epidermis with extended squid and primordia, EVI1 expression is greatly reduced and it is not detected in the structures. Here is the abbreviated form of eccrine sweat gland. Next slide.
So this slide is about the three methods whose results we'll be talking in the next slides. So first method is meta-analysis. It is a quantitative formal study design that is used to systematically assess the results of previous research to drive conclusions about modern research. It is a research process used to systematically and synthesize or merge the foundings of single independent studies using statistical methods to calculate an overall or absolute effect. Second method is gene enrichment analysis. It is a method to identify a class of genes or protein which is overrepresented, means the gene which has its proportion more than average, and if it is associated with these genes. Gene enrichment, it identifies the set of genes that are enriched in a particular data set when compared to a control genes. The third method is a network analysis. It's a process of investigating structure throughout the network of graph theory. It categorizes network structure in terms of nodes, ties, edges, links that connect. So to fully understand the genetic architect of a fingerprint, researchers perform meta-analysis on both East Asian ancestry cohorts and European ancestry cohorts using world and non-world phenotype of all temperatures. This meta-analysis includes 23,000 individuals, and they identified 42 independent signals mapping 105 genes. So in result, they found 4 signals were independently found in both East Asian and European cohort, and 27 signals were only found in East Asian cohorts, 9 of which were showing little significance in European cohorts. And in European cohorts, they only find 2 signals, and both of them were showing nominal significance in East Asian cohorts. So among the 18 East Asian cohorts which we have already found, so East Asian signals which we have already found through genome-wide association, 9 of them do not show any significance in European cohorts, but the rest of 9 they shows association with European cohorts and have some effect and have same effect as in East Asian cohorts. And these 10 signals were fully identified after meta-analysis. So the 9 signals which were found in East Asian cohorts and shows the same effect in European cohorts. These signals give proofs, and they shows that the fingerprint related genes are generally same in European and East Asian population with some differences that can be explained by the difference in frequency of genes present. So through genetic enrichment analysis, researchers found that the fingerprint-associated signals were significantly enriched for embryonic development, limb morphogenesis, and embryonic limb morphogenesis. In this figure, we can see the red color bars, which are associated with the limb development, and blue color bars which are associated with which are not associated with limb development, or they are associated with skin and epithelial morphogenesis. And we can also see the red star which shows the genes that are significantly enriched even after the threshold correction. Even enriched expression are also identified in mouse phenotype. Mouse limb tissues, distal fats, and foliage. It is clear that the blue color or the genes involved in skin and epithelial morphogenesis couldn't cross that threshold. It indicates that the fingerprint genes are related to the limb development genes. So, they further conducted network analysis and found that GWA's associated fingerprints were linked to the other proteins by known and regulatory functions. In this figure, we can see the circles. Circles and their circles indicate nodes, and nodes indicates proteins. And there are 2 types of nodes. One is some are empty and some are filled. And empty nodes represent the proteins that are independent of network, and the filled nodes represents the protein that are involved in regulatory interaction. So red nodes indicate the proteins involved in limb development, and the blue nodes are not involved in limb development. So there are red arrows. The red arrows, they indicate the 2 genes BMP. The red arrows, they indicate the 2 genes BMPFA and IRX3. As it has already been shown that these 2 genes guide in the phenotype of the middle 3 fingers. So this also shows that these 3 genes are these two genes and this pathway, these two genes and this pathway is a relevant explanation of the cause of pattern ground. So to further test the hypothesis that embryonic limb development may influence fingerprint patterns in limb-related phenotypes, hand phenotypes, hand phenotype, which is digit hand ratio, which is called DHR, was measured. It was found that the DHR is associated with finger patterns, for example, the frequency of whirl. Now, it was done in detail for the whirl type fingerprint. Here we have a graphical representation. So this is a graphical representation in which on the x-axis we have the whirl frequency of the 8 digits, and on the y-axis we have the DHR, which is the digit hand ratio. So it is clearly seen that for D5, which is shown in the red color, which is the red line, it is increasing linearly. That is, as the DHR is increasing, the whirl frequency is also increasing. Also for the D4, which is shown by the blue line, the relation is linear, whereas for D3 and D2, the relation is not very significant. So also it was found that the individuals with whirl pattern had 1.32 mm longer fifth digit as compared to the non-whirl pattern. So this was it for the DHR part. Now, apart from the individual digit length, it was also found that the phalanx length, the length of the distal phalanx also affected the whirl pattern. So the results provided evidences that fingerprint patterns are strongly influenced by the process of limb development. So this was the last experiment which was conducted. Now we move on to the conclusion. So here is the first conclusion. So the first conclusion says that there is a link between the height of volar pads and the type of fingerprint patterns, and the link was found to be consistent in all the experiments. So here is a summary that high volar pads lead to the whirl type pattern. Low volar pads lead to the arc type pattern, whereas asymmetric and intermediate height volar pads they lead to the loop type pattern.
Next slide. So this is the second conclusion. Principal function of the EVI1 is to promote proliferation. It is likely that altered EVI1 regulation changes patterns of cell growth and thus the length and shape of the distal limb and digit. As previously explained by me, EVI1 expression is found predominantly in the embryonic stage E11 and EVI1 is expressed in the mesenchyme of the distal ends of the digits under the volar pads, which later acts as the site of fingerprint formation. Next slide. This is the third conclusion. The proximal distal length proportions of hand, digit, and phalanx are established during the fetal period at the stage of fingerprint determination. So as Akanksha explained in slide 25, that higher the frequency of whirl pattern, longer is the digit length D5. Even the length of digits D2 and D3 were also associated with the whirl pattern. So here we come to the end of our presentation. Next slide.
So this is a picture. Yesterday we got a chance to meet Channa Basu ma'am. Audrey could not be present because she's out of town. So yes, that's about the picture.
We like and what we liked and learned about this Samyojana and about this paper. Also regarding paper, what we like, it's a very interesting topic, fingerprints, and what really interests us that how the gene which was found in a different experiment somehow end up related to the other morphogenesis in other part of the body. It's really interesting to know that the fingerprint pattern determines the length of your digit, and the methods used in the genome scan. It really fancies me, and I would like to study more about it. And what we like as a team as a process of this present process and experience we had while making this presentation and studying this paper. And the thing we learned in the Samyojana is accepting the feedback positively and to keep on implementing in the next presentation. That's it. Thank you, Anil sir.
Thank you, Akanksha, Sasya, and Adrija. I really like the way you ended in terms of the conclusions. I think I'm probably going to use the way you did that towards the end. So we have time for maybe 2 questions or comments. So the floor is open for the audience to chime in.
So you mute.
Okay, there you go. Sorry. The funny thing is I was talking about technical glitches. No, I wanted to commend your team, especially because despite a lot of technical challenges, you guys continue to be very involved in the process. So thanks for that. And any other questions or comments? Okay, if you have any questions as we go along, please don't, you can actually use the chat box and please don't hesitate to that. And I think we do have a few people on YouTube live as well. So if there are questions or comments there, you can put it there as well.
Sir?
Yes.
So yesterday we went to Chandana ma'am house and we invited her. So she couldn't attend, but her student Minal sir has attended our presentation today. So he's here.
Sorry. Oh, who is there? I mean, yeah, please invite him. I mean,
Shwetapal Mrinal sir is his student who works under Chandana ma'am. And today he has joined this symposium.
Oh, that's fantastic. Shwetapal Murnal. I'm very glad to see you here. So any comments or questions? Definitely comments, please. I'm not quite sure if Shwetapal Mrinal is here, I believe. So I'm glad he was able to join. and I'm sure you'll have some feedback from him in case he's unable to talk right now. And as I mentioned, any comments or
Sir,
questions?
actually, there is some network issue. He WhatsApp it, but he couldn't talk.
Yeah,
Because of network issue, he's not able to reach.
we just see his comment in the chat box. Nice presentation and no questions. Thank you, Mr. Manal. So we move on. I think I am very glad that we are sticking to our schedule. So we move on to the third presentation. Thanks again, Adrija, Sashya, and Akanksha.
Thank you, sir.
Thank you, sir.
Now we move to the third talk, maternal gut bacteria, CD4 plus T cells. Lalit. So while Lalit is setting it up, I am going to give you a little bit of background. So one of the key features of Samyojana is that we have to work in teams. And incidentally, for some reasons, unfortunate reasons, Lalit's teammates could not join in. I mean, well, they could not continue working on this the project, so they dropped in quite early. So Lalit had to do it by himself, and so kudos to his enthusiasm to have continued by himself. Lalit, the stage is yours.
Sir, am I audible?
You are. And
yes,
the
sir.
slides are, the slides are in place.
Okay, sir. So good morning, everyone. My name is Lalit, and I'm actually a first-year biotech student in the Vellore Institute of Technology. And today I'll be talking about this paper written by a bunch of scientists from MIT and Harvard. And through this paper, they were able to show that how infections in pregnant mice lead to neurological disorders and a dysregulated immune system in the offspring. So the brief, like to give you a brief idea about this paper, the idea behind this paper was that to show how children with autism spectrum disorders often display a dysregulated immune system, and to show that obviously they used a preclinical mouse model, and they were able to reveal the changes in the gut microbiome of pregnant mice and how it actually affects the activity of CD4 T cells in the offspring, which then leads to these immune prime phenotypes. We'll be talking about this. It sounds really complicated, but I hope that as we move forward with the presentation, things will get more clear. So here we have a normal case scenario where we have a pregnant dam who shows normal gut bacteria in its gut microbiome, and she gives birth to a normal offspring who shows normal behavior, and the accessibility of DNA between the histones is also normal. So the CD4 T cells, they show normal activity. And now we have the scenario here, which we'll be focusing on in this paper. That is, there's an inflammation during pregnancy. So here I've written that there's a maternal inflammation or maternal immune activation in the pregnant dam due to a pathogen. So what maternal immune activation basically means is that we're infecting the mother with a pathogen which activates her immune system. And since maternal is a word which we always use for when we're referring to the female counterpart, whenever we're carrying out an experiment, so we name the entire thing as maternal immune activation, which means activating the immune system in the mother. So since there's an immune response taking place, we'll have T cells everywhere, and inflammatory cytokines such as interleukin 17A, which is the one we'll be specifically focusing on in this paper. The interleukin 17A travels to the gut and starts destroying bacteria everywhere, since it'll recruit T cells to that spot, and then T cells will do their job. And then the same interleukin 17 then travels through the placenta into the embryo, the developing embryo, since we know that it's a pregnant dam. And then these alterations induced by interleukin 17A is seen in the offspring in the form of abnormal behavior and also an overactive immune system in our offspring. And to look at a molecular level, we can see that in comparison to the previous figure, more DNA is exposed between the histones. So the chromatin accessibility has been altered here, which means that there'll be an increase in the activity of T cells. we'll actually look at the experiments to prove how their activity is being increased as we move forward. So our hypothesis here is how changes in the maternal gut bacteria of the mother, obviously, drives intestinal inflammation in the offspring, which again is a keyword, along with neurodevelopmental disorders by altering the normal activity of CD4 T cells in the offspring. So I've broken down the hypothesis into these predictions, and in each prediction there'll be a topic, and then we'll go through the experiments they performed in the lab to try to mirror the results in the end, to show that what they actually assumed in the prediction turned out to be true. So we'll start with the first prediction. So our first prediction is the most basic idea, that how prenatal exposure to maternal inflammation, or MIA, we'll be using the word MIA for convenience. So how prenatal exposure to maternal inflammation increases susceptibility to bacteria-induced gut inflammation in the offspring. So our experiment starts with 2 pregnant dams. We inject the first dam with PBS, which is phosphate buffer solution, and it actually acts as a control experiment. There will be inflammation after injecting PBS in a pregnant dam, but it will be normal. We're trying to simulate normal inflammation in our first dam, while we inject our second dam with poly-IC. And what's different about poly-IC is that it's another chemical, but it does something else. It's able to actually mimic a viral infection. Since we can't actually use an actual pathogen, it'd be hard to regulate it. But when it comes to chemical, we can actually control the amount of chemical we're using to treat the dam, and in this way we can actually control the outcome. So by injecting poly-IC, we are able to achieve the MIA thing, or maternal immune activation. So after injecting our control with PBS and our target with poly-IC, they gave birth to these offsprings. You can see right here, and you can see that we're treating them with metronidazole. So why they did that was that they thought maybe after injecting them with PBS or poly-IC, they won't show the symptoms of the disease, like which will be infecting them with the bacteria afterwards, but they won't show the symptoms of the disease, since this commensal bacteria in the gut, and we know that it protects the host by preventing any foreign bacteria from growing in the gut. So now we have to take that out from the equation. So the best solution is now we treat the subjects with metronidazole, an antibiotic which we use normally in humans to treat infections in the stomach or peptic ulcers. So what this antibiotic does is it goes into the gut and starts destroying bacteria to reduce the diversity, which would then give the chance for a foreign bacteria to grow in there and recuperate. So after treating them with metronidazole, now we subject them to a Citrobacter rodentium infection, which is actually a well-studied bacterial model we use when we're working with mice or rats in the lab. So now that we have treated them with metronidazole, we have created the perfect environment for the bacteria to grow, the foreign bacteria. So after subjecting them to a Citrobacter rodentium infection, they started seeing symptoms of colitis, especially diarrhea and loss in their body weight. So now it was time to actually look what's happening on the inside. And they performed a colon histology as well as immune cell analysis. And what they found is what you can see here. They saw a reduction in the colon length, and I've actually drawn this line to compare the reduction in the size of the colon. The one on the right is the colon which has been taken from an MI individual, and on the left is the one we have taken from a PBS individual. And you can see with the line I've drawn that there has been a reduction in the size of the colon, which is normal because of the collateral damage caused by an immune response. There's going to be cell death, and it's obvious that the size will decrease. And then there's also inflammation we can see here. This is like a cross section, a side view of the colon. And again, I've drawn a line here to show that there's been an increased inflammation on the part of the MI individual. This is how normal inflammation is supposed to look, while you can see a severe form of inflammation when it comes to the MI individual. So from now on, I'll be showing only the figures. So just to be sure that you have an idea what I'm talking about, just keep this image in your mind whenever I'm talking about the colon length or inflammation. I won't be showing these images again and again. I'll be showing you only the figures. So just so you're not imagining things in your head. Okay. So now coming to the part where they analyze the immune cells of the individuals. And we'll be focusing on only 3 particular things The first one being here percentage of interleukin 17A, which is actually the suspect, our main suspect in this paper. And the other one will be the percentage of interferon gamma. Again, another inflammatory cytokine. And the function of these cytokines is to recruit T cells to the spot of infection. So if you look at the figure on the left, you can see these clusters, and you'll be wondering what these clusters are. Since we know that whenever we're performing an experiment in the lab, we don't just stick to 2 test subjects. There are multiples and n number of test subjects just to be sure that the data is actually producing the same results. So the clusters here represent the average results we get. And the ones here on the top, they're actually exceptions. So we won't be focusing on the exceptions. If there's an experiment, obviously there will be exceptions. But we won't be focusing on the exceptions. We'll be only looking at this cluster right here. So you don't have to even worry about that. Just look at those peaks right here. That's the main thing. And so coming back to the part where on the x-axis we have the peak for the PBS offspring and the MIA offspring, and right here we have the percentage for interleukin 17A T cells. So we can see that there's an increase in the percentage of interleukin 17A in the MIA offspring in comparison to the PBS offspring. The same can be said for the percentage of interferon gamma, again another inflammatory cytokine, and the percentage of interferon gamma is more in the MIA individual in comparison to the PBS. And we can conclude from this that an increase in interleukin 17A and interferon gamma actually results in an increase in inflammation. So just keep this in mind that we'll be only referring to these 2 inflammatory cytokines. And the third factor I talked about earlier was FOXP3 CD4 T cells. And FOXP3 CD4 T cells sounds really fancy, but it's just a type of CD4 T cells which has a regulatory function. They're basically called regulatory T cells, and their function is to bring back the immune response back to normal, to like slow it down and then bring back the homeostasis in the immune system. So again, if these things are to be positively regulated for an increase in inflammation, this thing has to be like same. This has to be like there should be a positive regulation for the regulation part as well in order to bring back the immune system back to normal. So if we look here, we see that the percentage of T cells which are regulating the immune response, it's higher in the PBS individual in comparison to the MI individual. So holistically, we can see that there's more inflammation. The conditions which are increasing inflammation in the MI individual are more, while when it comes to the PBS, we have less of inflammation and we have more regulation. So that we'll be only referring to the 3 of these here: interleukin 17A, interferon gamma, and then FOXP3 cells. So just keep a note of it. So now that we have a basic idea of how everything is happening, we will now look into the specifics of it. So our next prediction is that the behavioral abnormalities in the MIA individual, our target individual, are determined prenatally, which means before birth, while the immune prime phenotypes, which is the gut ailments, they arise after birth. To show that they had a different approach in this experiment, we call this a cross-fostering experiment. And what we do here is we have 4 different pregnant dams, one being PBS, poly-IC, PBS, and poly-IC. And what we do here is we allow the dams to give birth to offspring, and then we switch them after birth, just to see. This is what the cross-fostering experiment is all about. So we switch the pups after birth, and you can follow these arrows right here. For example, let's just take this one example. We have a PBS dam here. She gives birth to these pups, but we're actually not allowing her to foster her own pups. We're actually giving her the pups of another poly-IC dam. So in this way, we actually get 4 combinations. I'll just explain one of them, which is a PBS offspring being fostered by PBS dam, and then my offspring being raised by a PBS dam. In a way, we get 4 of these combinations. So before moving on with the actual figures and all the numbers, I'd like to give you an intro about the test they perform in the lab in order to figure out if there's something wrong with our individual, like if it's actually suffering from any of the sociability deficits or neurological disorders. There were 2 such assays, and I'll try to stay as brief as possible to give you a really nice idea. I'll add pictures here. So the first assay is called the three-chamber assay test, and they performed it to see whether the test subject has a social preference. And what they do here is there's this system of three chambers. You can't see it here, but they're actually interconnected. And we place a test subject in the middle chamber for the first time, and we let it explore the setup for say 15 minutes. And then we take the setup apart. We set it up again, but this time we place two cages on the right chamber and the left chamber. The left chamber is empty, while the one on the right has a stranger mouse. And now again we let our test subject explore the system. So this time we have something new here. We have a stranger mouse in the right chamber, and we see if our test subject tries to initiate contact with the stranger mouse. And depending on the number of interactions between a test subject and the stranger mouse, and how much time it spends away from the stranger mouse, or spends more time in the chamber where there's no one, we can actually compute the data to see if our individual shows normal social behavior, or is actually suffering from any sort of like autistic disease. So after performing this test, they came up with this this figure which you can see here, and it's more simplified right now. We have a PBS and an MIA offspring being raised by a PBS dam, being fostered by a PBS mother, and then again on the right side we have a PBS, the same PBS and MIA offsprings being fostered by a MIA dam. And there's two peaks for each individual. The first peak, you can see they are filled circles. They're actually for the social targets, and the social target is stranger mouse, right? And then the ones which are not filled and are empty circles, they are actually the inanimate targets, by which they mean the empty cage. So let's just compare the two offsprings being raised by a PBS dam. We can see if we just compare these two peaks for social targets, we can see that the PBS individual prefers the social targets over the inanimate targets. By comparing the two peaks for social targets, we can see the peak is higher, taller for the PBS individual compared to the MI individual. And on the other hand, our MI individual prefers the empty cage or the inanimate target over when we're comparing it with the PBS individual. And we can see the same trend with the MIA, the offsprings being fostered by the MIA dam. We see that again our PBS individual prefers social targets over inanimate target, while our MIA individual prefers to stay alone rather than try to interact with the stranger mouse. So the next assay performed is called the marble burying test, and the reason they performed this test was to see if the test subject shows any sort of repetitive behavior. And how they were able to demonstrate is that demonstrate that fact was that they came up with this different setup where they filled the a closed system with sand, and they place these marbles on top of the sand, and then they allow a test subject right here to explore the system. And they noticed that the subject was burying the marbles in the sand. Okay, and then the next time they take the setup apart, and every time they were digging those marbles out and then placing them again on top of the sand, the subject was again and again repeating the behavior of burying the marbles. So that's why we have the name marble burying test. And we know that if the test subject is repeating a behavior, there's something wrong with the test subject, some sort of OCD-like behavior. And with that, they were able to come up with this figure, where again we have the same stats with the previous graph: a PBS and MI dam being raised by PBS dam, and then a PBS and MI individual being raised by an MI dam. And we can see that the individuals being raised by the PBS dam, specifically the MI individual, shows more repetitive behavior, while the same trend is seen here when the same individuals are being raised by an MI dam. Again, we see that the MI dam is showing more repetitive behavior in comparison to the PBS offspring. Now that we've seen the behavior part, we come to the histology results, and we see here there's no difference between the reduction in colon. Earlier we had a figure, and now we're just having the graphs right here. So I'll just assume that you know what I'm talking about here. And if we compare the two individuals being raised by PBS dam, the colon length remains the same. And again, when we're talking about the individuals being raised by an MIA dam, the colon length remains the same. There's no drastic difference between the two individuals. But when when we come to the part where we're analyzing the immune cells, again, you might be seeing a big difference between when it comes to the individuals being raised by different dams, but that doesn't matter. We're just comparing two individuals being raised by the same dam. That's what we're bothered about. So if we look here, we can see again no drastic difference they're being raised by PBS dam. Same with MI dam. We compared both the peaks, no difference. Same here. When we're talking about the percentage of interferon gamma. Also, when we're talking about FOXP3 cells, we can see that there's a slight difference in the peaks, but I could say that they're not really explained why we can see this difference. So we summarize from the experiments that it doesn't matter who fosters the offspring. If they're genetically related to the MIA dam, they will suffer from behavioral abnormalities, hinting towards its prenatal origin. While the gut ailments we're seeing, they originate after birth and are only seen in those individuals who are being raised by the MIA dams. So the next thing we're looking at is how inheriting the altered gut microbiome. What's the role of the gut microbiome, and that how it's sufficient enough. Inheriting the gut microbiome is sufficient enough to produce just the immune prime phenotypes. So they had a different take while performing this experiment, and now that they had to isolate just the gut microbiome of the pregnant dam, since we can't just literally take the gut microbiome and transfer it to another individual. The next best alternative is to transfer the fecal slurry, which is actually a part of the gut microbiome, from a PBS dam, like it's written here, to a germ-free female, and then the fecal slurry from an MIA dam to a germ-free female, and they mated both of them with different germ-free males, and they gave birth to a PBST offspring and an MIST offspring, where ST stands for stool transfer. And they again subjected them to the social preference test. I've added an image so that you have an idea, a brief idea of what I'm talking about here. And we can see here that they don't show any, there's no difference. If you compare the two peaks for social targets, they're pretty much the same. And then for the inanimate targets, again, they're same. And talking about the marble burying test, no significant difference between both the subjects. They almost show the same normal behavior. But when it came to the comparison of colon length, we can see that there's a reduction in the colon length of the MIST offspring in comparison to the PBST offspring. And again, when we came to the part where they analyzed the immune cells, they saw that the percentage of interleukin 17A was shooting up all the way up in the MIST offspring in comparison to the PBS or our control offspring. And then again, we see the same trend, the percentage of interferon gamma, more in the MIST offspring. And conversely, on the opposite side of the spectrum, we see that the percentage of the cells which regulate the immune response they have decreased in their population in the MIA individual in comparison to a PBST individual. So these data collectively support the idea that an altered gut microbial community, rather than a bacterial species capable of supporting a specific T cell response in pregnant mice exposed to inflammation or MIA, is both sufficient and necessary for the emergence of a long-lasting immune prime phenotype in the MIA offspring. So coming now that we've looked at an overview of what's happening on the outside, we'll now look at what's happening on a cellular level, on a molecular level. So our first prediction, starting off from here, is that what's the role of T cells, and that how MIA-CD4 T cells are preferentially, specifically being differentiated and activated, and they are leading to all of these manifestations we are seeing. So to show that, we have to specifically focus on the T cells, and they started with again with a PBS offspring. They carried out all those experiments earlier, and now we're just to reduce the confusion, we're actually starting with a PBS and an MIA offspring, and they're extracting naive CD4 T cells from the PBS offspring and then the MIA offspring, and they're transferring it into two separate RAG1 deficient mouse. And now what's RAG1? RAG1 is actually a pathway in mice which is responsible for the production of T cells in the individual. And if it's deficient in a subject, which means that there won't be any T cells, and they're actually immunocompromised. They don't have any T cells in their body. So again, they're immunocompromised, and by transferring these naive CD4 T cells into their bodies, those T cells would realize that there are no T cells around, and now they have to like recuperate and bring back their numbers, and they'll start proliferating. And after a few weeks, they actually started seeing like right here, if you see on the x-axis, we have the weeks, the duration of weeks after transfer, and on the y-axis we have the body weight percentage. So now that we know that if there's going to be if the test subject will suffer from diarrhea or anything remotely related to colitis, they'll lose weight and they will start looking sick. So after the third week of transfer of those T cells, we see that the individual who received the T cells from the PBS offspring didn't notice any sort of body weight drop in. But when we're talking about the individual, the RAG1 individual who received the MI T cells, they started losing weight. As you can see here, that the body weight of the MI offspring drops. And again, it was obvious that they had to check for the colon length and the inflammation of the colon. And they can see that the individual who received the MIA T cells, there was a reduction in the colon length of that individual in comparison to the PBS or the control experiment. And again, you can see here that this part on the top was a side view of the gut of the PBS or the control experiment, and this seems pretty normal. And this here we can see there has been inflammation in the MI individual. So just to be sure, they performed another experiment to test the thing they found in the previous experiment. What they did was that they extracted naive CD4 T cells from a PBS offspring and then an MI offspring, and then mixed them up in a 1:1 ratio together. And they made sure that they tagged this, the MIA CD4 T cells, just to be sure later on when they separate that they will be able to easily separate those cells for analysis. And after mixing them up in a 1:1 ratio, they transferred again them into RAG1 deficient mouse, waited for a few weeks so that those T cells can get some time to proliferate, and then they extracted those T cells, and they were able to easily differentiate those cells since they had tagged the cells separately with markers. So now we have the blue ones here, which are the CD4 T cells from the PBS offspring, and then the orange ones from the MI offspring. And then they subjected them to RNA sequencing. So the RNA sequencing actually gives us this chart-like thing called the gene set enrichment analysis. And you can see I've given an example here. It's not related to the info I'll be telling you, but even I don't know how this works, but I'll just try my best to explain you what I understood while like trying to read about how this enrichment score thing works. So how this works is actually we can see this peak-like thing which is shooting up right here, and you can see that there's a red portion here and a blue portion here. So it's like a spectrum right here, and the red portion actually represents our target individual, while the blue one represents a control experiment. And these barcode-like things you see here, they're actually the genes. So by seeing that this peak shoots up at the red part, and then it starts gradually decreasing when we move to the blue part, which means that these genes, these barcodes you see here, they are being upregulated. While when it comes to the control experiment, it's back to normal. So by using something like this, a chart like this, which they got after sequencing, RNA sequencing, they found these three things to be upregulated. The first one being mitogen activated protein kinase, which is called MAPK, and its function is to relay and amplify cell proliferation in an immune response. The second thing being interleukin-6 JAK-STAT3 pathway, which again regulates cell proliferation and differentiation. And the third one being an overall increase in differentiation of Th17 cells. So at this point, you can just figure out for yourself that all these three things have one thing in common, that all of them are somehow amplifying cell proliferation and immune response. So this explains that why we are seeing a heightened immune response when it comes to an individual who's MIA or has actually received MIA CD4 T cells. So the next thing we'll be looking at is how these MIA CD4 T cells are undergoing epigenetic changes. Remember that that histone and DNA structure I was showing you in the beginning of the presentation, how there's been a difference in chromatin accessibility. We'll be actually exploring that part in this prediction, where we'll be showing how they performed experiments to show that they were actually being epigenetically primed to become hyperactive. So in this experiment, again, they started off with a PBS offspring and an MI offspring. They extracted naive CD4 T cells, and they subjected both of them separately to attack sequencing. And attack sequencing is a method in which we have these small enzymes you see here, these small lines you see here. They actually an enzyme named translocase. And what it does, it binds to the exposed regions between the histones and then cuts the DNA off. And we can actually analyze the length of the DNA. So the more DNA, the longer the piece of DNA is, the more upregulated the part of the gene is. And there'll be more transcription and hence more activity. So the first thing they found to be upregulated was this gene named BRD4. It stands for bromodomain 4. And again, the function is really obvious. It increases the acetylation of histones. So it's like sort of upregulating the activity of the T cells. And you can see it's obvious from here that the relative mRNA expression of this gene is more in the MIA T cells in comparison to the PBS T cells. Again, another gene named interleukin 6, responsible for cell proliferation. It's related to the interleukin family, and it's more in the MIA T cell in comparison to the PBS T cell. The expression, relative mRNA expression. And then finally, we have the MAP4K2. Again, belongs to the mitogen activated protein kinase family. And it's again responsible for regulation of T cell differentiation. So if there's more relative mRNA expression again for this gene in the MIA T cells. So again, collectively, we can say that in the MIA T cells, there's been like an upregulation only of those genes, which would increase the activity of T cells. Again, a reason why we are seeing a heightened immune response. So finally, we'll come to the part where we're talking about interleukin 17A, which was actually a primary suspect in the entire thing where actually in all of what they performed in the lab and then what they wrote in this paper, interleukin 17A was the most crucial thing they actually talked about in the paper, and it was actually the primary suspect. So we'll see that how maternally induced interleukin 17A is critical for immune susceptibility in the offspring. So in this experiment, they took 3 different dams, and they injected the first dam with immunoglobulin G. And again, immunoglobulin G has a really crucial function when it comes to an immune response in mammals. So they injected the first dam with immunoglobulin G, and later on with PBS, and then again another dam with immunoglobulin G, but this time with poly-IC. But the third time they injected it with something different, with anti-interleukin 17A, and then with poly-IC. So the only thing you have to like keep in your mind when we're talking about these 3 pregnant dams is that the most unique one is the third one. Why? Because since we're treating it with anti-interleukin 17A, the body of this particular subject won't contain any interleukin 17, right? While the other 2 will have. So that just keep that in your mind that one of them is missing interleukin 17A, while the other 2 they have interleukin 17A. And then they name the offsprings depending on what they were injected with, like PPS isotype. And then since we're injected with poly-IC, we have the name MIA, and then again the isotype thing. And then we have something different here since it is an MIA offspring, but it doesn't have anti-interleukin 17 in its body. So when we were comparing the colon length of all those 3 offsprings, we see here that the PBS isotype and the individual which doesn't have interleukin 17 in its body, they have comparable colon lengths, while the individual which still has interleukin 17 in its body, there's been a reduction in its colon length. So that's a really weird thing happening here. And just to be sure that what we're hypothesizing here is correct, they analyzed the immune cells of all those 3 offsprings, and we actually could see the trend here right here that after seeing a reduction in colon length of the MI isotype, we again see here that the percentage of interleukin 17A is obviously comparable between the control experiment and the one which lacked interleukin 17A in its body. While again, we see an increase in the percentage of interleukin 17A in our MIS type individual. Again, the percentage of interferon gamma is way ahead in the MIA type individual in comparison to the other two. And obviously, the percentage of FOXP3 cells or the regulatory T cells is decreased. The percentage it's decreased in the MIA isotype in comparison to the PBS isotype at control and the anti-interleukin 17 individual. So we summarize from this that interleukin 17A induced changes in the maternal gut microbiome. The immune and the behavioral phenotypes observed in the MIA offspring depend on the cytokine interleukin 17. And that changes in the maternal microbiota was sufficient enough postnatally to produce inflammatory phenotypes. But on the other hand, the behavioral abnormalities of the MIA offspring require the expression of interleukin 17A receptor in the embryonic brain and its activation during the prenatal period. So now that we have idea of a basic idea of what's going on, we'll not talk about therapeutic application they came up with while they were writing this paper. So now that we know that interleukin 17A is really crucial behind the origin of all these manifestations, everywhere we see that interleukin 17 is always there, and by removing interleukin 17A, like in the previous experiment, we see that we don't see any of those disorders manifesting in the individuals. And like even a normal person could say that obviously if we could block the synthesis of interleukin 17A, we can prevent any of this happening. And this was the idea they came up with, that by blocking interleukin 17A in pregnant mothers, who we're actually paralleling now it with humans. It's not about mice anymore. So now with that we're talking about humans, by blocking the activity of interleukin 17A in pregnant mothers who actually, for example, have undergone any sort of infection or are vulnerable to any future infections during the term of pregnancy, we can actually prevent these diseases from arising or manifesting in the children, like the autism part or the gastrointestinal ailments. We can actually prevent them from happening. And overall, the study provides an insight into how environmental stimuli, especially maternal inflammation, could dysregulate the offspring's immune system and then render the particular individual vulnerable to inflammatory challenges later in life. And more specifically, it offered important insights into the occurrence of inflammatory dysfunctions that often accompany, say, neurodevelopmental disorders. So this paper actually gives us an idea of how we can approach a cure for autism, which is a big problem, say in India or even globally, where thousands and thousands of children are suffering from autism, and that if we can actually apply this idea to work towards a solution to severe the causative thing, which is actually giving rise to these diseases before the child is being born, so that we don't have to worry about them after the kid is born. So that's a really nice idea, and it's not been practically applied again, but it's a really decent solution for trying to tackle diseases like autism in real life. So with this, my presentation comes to an end, and thank you for your attention. And now I'll talk about what actually caught my attention in this paper. It was actually, when I actually started reading this paper, I was really cynical about it. I thought maybe it's really boring, but when I actually started reading it, they were actually making these references to a theory they were talking about, and they actually assume that you must be knowing it, since that's what the paper is based on. And I found this theory which involves a system involving the gut and the brain, and that there's actually a bidirectional communication system between what's happening in the gut and what's happening in the brain. So what was actually peculiar was that two remotely related systems, like what's happening in your central nervous system up in the head, and then what's happening in your gut, they're even related to each other. It's a really weird thing, and it actually opened so many doors for me. I started reading here, and then eventually went to read more about that theory. It's not really as of now they've not proved it, but it's a really nice theory. And and then talking about what I actually learned from this entire program is that earlier I used to like read only books, and then I never went for research papers. Since whenever I looked at those figures and those numbers, I used to get scared, and I would be like, it's too complicated for me, and I should avoid it. And I used to stick to textbooks. So after actually going through this program and that making a presentation, trying to understand all those graphs and stuff, and I've actually switched. I have a preference for reading research papers now, since research papers actually they give you the evidence, they put forth the experiments, and they give you room to think yourselves, rather than those textbooks. They just put the science in front of you and they just assume that yeah, you can just learn all of it. So that's a really nice approach, and I'll thank sir for actually giving me this this opportunity to become part of this program, and I got to learn a lot from sir, and yeah, that's it. Thank you.
Thank you, Lalit. Very nice. As I've always enjoyed our interactions and learning, especially about this particular story. So we have time for maybe one comment and a question or something. So anybody in the audience? I know Bhavani, you plan to leave. Do you want to? So we come to the close of the first session. So any comments broadly for the first 3 talks, suggestions?
Hello. Yeah, hi, I'm Bhavani. I'm sorry, I had to leave midway for the second presentation by Akanksha, Shashya, and Audrya. But whatever I heard, I liked it. And so that's why I could not comment much on that. With the third, Lalit, it was amazing. It was so good. Lalit, I really enjoyed the talk, and you were very clear. Your words were clear, and you explained it so well that I could follow it very well. Only thing is, and even the science is very interesting, right? The paper is very interesting. The thing is that you talked about ataxia. The ataxia, sometimes we have to be really careful because you've been so good with the presentation. I just want to say ataxia talks about the open chromatin, but open chromatin does not automatically correspond to gene expression. So you didn't show that much about that. And overall, the presentation was very nice. The only thing I would have liked is to have like a header near the result. So when you're explaining, we actually know what the result is. So it would be a little bit easier to grasp. But I felt you did an amazing job. And I enjoyed all 3 presentations. The first presentation and third, I watched fully. I listened to fully, so I understood it much better. And all the very best to all of you. You're doing a very good job, much better than what we used to do when we were undergrads. So thank you, Anil, for this opportunity. And anytime if you need any help, to write to, I'm a research associate in New York University, Abu Dhabi. I studied in the US. Actually, I started at Ohio State, where Anil also studied PhD. We met in Abu Dhabi, but we have a common connection. So if you need anything, or if I can be of any help, you can please write to me. Thank you.
Thanks, Bhavani. And I'm looking forward to our continued interaction, especially in this project. And maybe we can make it widely accessible to undergrad students. And we'll talk more about that. And again, we would love to have you back as the day progresses, but I do know that you have other responsibilities. So if you can hop in, please do. And we'll continue our interactions. Thanks again. Bye. So any other questions? Or I think we've come to a conclusion of the first session. And I'm very happy that we have our next guest, Dr. Sudha Rajamani from IISER Pune. So welcome, Sudha. I am...
Hey, thanks. Hello,
good morning.
Hello. Morning.
So we have a short break. If you guys want to go stretch your feet, we'll start in about 5-7 minutes. So Srishti and Sarah, do you want to get set up?
Yes, sir. We'll try sharing the screen once.
And since Sudha ma'am is already here, we don't have to wait till 11:20. If you're all ready, just give me, if you're taking a short break, let me know. And whenever you're ready, we'll get started.
I hope the screen is visible to everyone.
The screen is visible. It's not a slideshow yet. Yes, it is now.
Let me know when to start.
Yes,
I hope Sarah is here too.
I believe so. So do we have a, so does anyone have an objection if we start at 11:15 instead of 11:20? If you have an objection, or maybe I should ask if you're all okay, raise your hand. It doesn't usually happen in symposia where we start early. Typically, it's always a few minutes late. We have another guest, which is so Srishti. Dr. Krishnamurti is also here, which is fantastic.
It's great. Hello, Soren. Thank you so much for coming.
Hi, Ram. What a surprise.
Oh, hi. I just got this, invite, and I wasn't sure whether I will be able to join, but I can. I've taken a break from the kids for a while, so yes, I can. Let me switch on my video. I hope you can see me.
Wonderful to see
Hi.
you. Hi.
Yeah.
So, Srishti, can you let me see? So it's actually a privilege and an amazing opportunity for us to have two leading researchers in this field. So Dr. Krishnamurti, I know you had your video on.
Oh, yeah. My video is on, so don't worry. Yeah.
So this was specifically an interest of, this was motivated by Srishti's interest in astrobiology, I believe. So she kind of insisted me, and since I know Sudha, I was able to twist her arm and make her come here. And I'm actually delighted that Dr. Krishnamurti is also able to come. So just since you are all, you have come for the first time, I just wanted to briefly tell you about Samyojana. And the reason I'm going to tell you about this is to get your inputs about this effort. So Samyojana is an undergraduate student-centered initiative. We are trying to be as pan-India as possible, and it is actually growing by the word of mouth. The main focus is to encourage reading, presenting, and discussing primary research literature amongst undergraduate students. And more, I'm trying to make an effort to make it happen early in the undergrad the study program than later. As I kind of alluded to, the aims are enabling critical thinking and analysis, instilling professional presentation skills, and fostering collaboration and teamwork. So I'll cut to this chase. So since 2020 August, we started this in Hyderabad, but it spread by word of mouth, and now we have students from many cities in India. And over the last 2 years, we've been testing different formats on how to make this much more effective. And the symposium is our newest experiment. So thanks again for being here. It is a nice, it's 11:17. And since Dr. Krishnamurti is in California far away, and I don't want it to be too late for him, shall we get started? Srishti and Sarah.
Yes, sir. I'll share my screen. I hope again it is visible and I'm audible clearly.
You are.
Okay. So thank you so much, sir. Hello, everybody. I'm Srishti Chipre along with my teammate Sarah, and I welcome you all to the molecular biology section of the Samyojana Symposium. Today, we are going to present a paper titled A Plausible Prebiotic Selection of Ribose for RNA Formation, Dynamic Isolation, and Nucleotide Synthesis based on metal-doped clays. Or in other words, why is ribose the chosen one? So before we get into this magical drama, we first have to make peace with the fact that man has always been and will always be curious about his or her origins. Because since we have been given this tricky little thinking brain, we will continue to seek justifications and answers regarding our origins. And one way to do that is to just observe the world around us, gather the information that we have, make assumptions on its basis, and then trace back into time to seek for evidences. And this art of scientific exploration was mastered by Charles Darwin. He used to say that there is grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity from so simple a beginning, endless forms and most beautiful and wonderful have been and are being evolved. So it is indeed very fascinating to see that so many different forms, diverse forms and species persist today. And yet in the network of life, we share at least one common ancestor. And this beautiful mess of life has been given rise by this tiny little cell called the protist. Now, Darwin didn't talk about the protist itself or how it came into being, but that will be our topic of interest for today. We're not interested in what happened after life came into being, but how exactly life came into being on Earth, or what exactly is the recipe for life. When we talk about recipe, a perfect recipe is not just made from the alignment of different situations and conditions, but the ingredients are also very important. And as a biotechnology student, when we say life, we say biomolecules. So there are these different biomolecules, proteins and nucleic acids and lipids and carbohydrates that maintain this whole system of life. But since we are looking forward to seeking some answers to our questions regarding our origins, we can look right here in this molecule which stores all our genetic information, the nucleic acids. And in order to study or understand the nucleic acids, we first need to understand its structures. Apart from the phosphates and the nucleobases, the most important component of nucleic acids is the sugar. So these are the two sugars present in both the nucleic acids, the deoxyribose sugar and the ribose sugar. And amongst the two, the ribose is very important because it is the precursor of deoxyribose to construct DNA and also the essential backbone of RNA, which is believed to be the first genetic polymer for the emergence of life. So the next question that comes to the mind is, why ribose and why not any other sugar? Why is ribose so exclusive to RNA? Another question that you might ask is, aren't there any questions before this that need to be tackled or any gaps to be filled? The straightforward answer to that is yes, of course, there are many questions, but we are just trying to answer one in this sea of questions and just trying to fill one gap. And the question is, why is ribose the exclusive sugar for RNA? And through the paper, we can also see what could be a plausible solution for the RNA problem. So let's just first focus on what exactly is the RNA problem and what is the solution that the paper is trying to present.
So we have the question here: why is RNA exclusively the sugar used in RNA? So the main issues that we face when we are talking about selecting a sugar for the nucleic acids involved and talking about how ribose was specifically chosen is that most of the reactions that are known right now for either the synthesis of sugars or the conversion of sugars to nucleic acids, they are unselective, as in they don't specifically choose ribose over others. They lead to many unstable intermediates, and even the ribose synthesized after, we don't really know how it was stabilized for long periods of time, because it's not necessary that immediately after the synthesis of ribose you immediately get the next step into synthesizing RNA. And usually there are the yields involved with the synthesis of ribose are pretty slow. So what this paper particularly suggests is that instead of having like a very specific sugar forming reaction, what they suggest is that once the sugars are formed, there are very specific minerals called metal doped clay. We'll come back to the terms a little later, which bind to ribose specifically, selectively. And through this, there is a separation that occurs. Thereby, we isolate and accumulate ribose, which then we could use later on to form nucleosides and therefore RNA and so on and so forth. So that is what this particular paper suggests. So, so there are two particular ways where that the paper explores. One, you start with the possibility of or a mixture of sugars. We have specific chemical reactions that selectively chooses ribose over other sugars involved. And then we get a ribose-rich environment for the formation of nucleic acids. The other option they suggest is we start with a mixture of sugars again. We separate ribose through various techniques, could be physical. And then we get the same thing. We get a ribose-rich environment for the formation of nucleic acids. The end result here is that ribose specifically becomes chosen for this further reactions to RNA and DNA and so forth. And there have been previous studies that kind of look at all of this. How specifically ribose was chosen. And they usually follow the first, or try to follow the first approach. Benner and group suggested the borate hypothesis, and other groups kind of tried to study the details and see if it would actually work. There's also a silicate hypothesis by Lambert and group. And there have been multiple studies on this and computational studies involved in the calculation of the energies and the stabilities to see how well these theories hold. And there are some positives and negatives, as always. So coming to the borate hypothesis first. So borate is found to be effective in stabilizing ribose against degradation over other pentoses. So this would mean that ribose specifically is isolated, and the Lambert, the other group kind of calculated the energies involved, and they found that for like a borate ribose complex with a 2:1 ratio, ribose is the most stable. And the only issue with this is that boron is not very abundant in the Earth's crust. So you don't have a lot of boron. And for the amounts required, it would completely change what we know about the early Earth surface. And there's an image of how boron would theoretically stabilize ribose or any other pentose sugar. Going on to the silicates. So the Lambert group suggested silicates can mediate within the foremost reaction and therefore stabilize sugar products. But the issue is that all the studies done till now, they couldn't really say if ribose is particularly favored. They just said that sugar products are stabilized. There was no indication that it is specifically ribose that was stabilized. And before we move on now, we have to talk a little about the specific terms that keep coming up again and again in the paper. And so the first one is ligand exchange chromatography. So the ligand exchange chromatography is a chromatographic technique. The stationary phase involved is a cation exchange resin. And usually on the resin, there are a lot of metal ions that are loaded. And there could be magnesium, calcium, zinc, sodium, a lot of those guys. And they tend to form coordinative bonds with the molecules from the mobile phase, and usually are analytes. And the elution is done, and the separation is based on the differences of how strongly each analyte interacts with the metal ion and the resin. So if an analyte interacts very strongly with a particular metal ion, it takes a lot more time to go through the column than something that doesn't interact as strongly. And through this innate differences in chemical reactivity, you could ideally end up separating a lot of these analytes. So that's about ligand exchange chromatography. The other term that comes up a lot, especially when you talk about chromatography, is the retention factor. So the retention factor can be thought of as a measure of how strongly the compound or the analyte interacts with the stationary phase. And you could think of it as sort of, in the simpler TLC, you'd have a stationary phase being the thin layer and the mobile phase being the solvent. And as your sample is loaded, and the more the sample interacts with the thin layer, the slower it takes, it travels up through with the solvent. And so the stronger a particular sample interacts with the given stationary phase, the larger the retention factor. Keep in mind that a retention factor is entirely based on the molecule itself and the chemical species involved, and it does not change unless you change either the stationary phase or the molecule. The third thing that keeps coming up a lot, and I won't spend too much time on, is the Formose reaction. The Formose reaction is a very generalized sugar forming reaction. So it was proposed, and you usually take formaldehyde, and at the end of the reaction, what you get are a mixture of pentoses, hexoses, tetroses, and there is no specificity involved to say that, okay, you get ribose isolated off from this. There have been many catalysts suggested that could ideally get ribose, but then the reaction conditions, the catalysts involved, all of that are so very specific. You can't really expect something like that to happen spontaneously in nature in large enough quantities so that you can get like RNA separately. So the Formose reaction is this general reaction to synthesize sugars, a sugar forming reaction, basically to say. The last term that we'll be looking at for now is metal doping. In metallurgy, mostly, metal doping means the addition of a second metal or a mineral in a very small quantity to a huge amount of another metal or a mineral. So you'd see a lot of this in semiconductors and stuff, where you add a small amount of a secondary metal to like silicon or germanium to get a very improved semiconductor. And similar properties can be done, can be created for clays and other minerals, where addition of a small amount of metal changes certain properties that are required for us. So you're basically introducing impurities to improve the properties that you're looking at. So that's about metal doping.
Yeah, so as Sarah has already mentioned, there were two approaches mentioned in the paper. One was that we could look at the pathways, the sugar forming reactions, and see whether any reaction is yielding ribose specifically. But the basic idea in this paper was that if we have a mixture of sugar and we can easily, feasibly separate ribose from that mixture, we can actually get the answer to a question of why ribose is so favorable for RNA. So what they did was they performed a few experiments to test the retention factors of various sugars, so that we know that it is not biased on our end to ribose, the experiment, and it is also testing the other sugars. So basically, in all the experiments that they performed, whether they tested in different columns, it was found that only ribose has some significant retention activity. And this tells that we can move ahead with this idea. So the retention degree of solutes was measured by the retention time, volume of the eluent, or the effect of the solute distribution on the stationary phase. Now that we have some significant data, we know that ribose has some significant retention activities when performed in modern separation techniques like ion ligand exchange chromatography. The next step is to make the experiment a little more relevant to the prebiotic era. And for that, we first have to find a natural environment that can act as a stationary phase. So here they are giving us two established facts. One, that the early Earth was a rocky planet, and that is why clay minerals could be a suitable choice. And the second one, that these clay minerals have some cation exchange capabilities. So the clay mineral specifically used in most of these reactions is kaolinite because of its distribution around the globe. And since the ligand exchange chromatography requires metal cation bound to the stationary phase, we can envision that a clay mineral exchanged or adsorbed with a metal cation might be regarded as a natural analog to the ligand exchange chromatography system, and it should be capable of separating carbohydrates produced in formose or other analogous reactions. So now we have established our stationary phase in the natural environment, that is a clay mineral adsorbed with a metal cation, and that is the metal doped clay systems. So next up, what they did was they analyzed different sugar mixtures using IR spectroscopy, for the simple reason that IR spectroscopy is sensitive to absorb the retention activities or adsorption activities of small molecules. And quickly, the way this works is that we have an IR source and we irradiate the light from the IR source to in a given sample. It either absorbs some of the light or transmits. So the transmitted light can be detected using a detector, which will give us our IR spectra. And this is how the system looks like. So a graph of the IR spectra will look something like this. The wave numbers basically is a measure of frequency. Transmittance, as I already told, is the measure of how much light is transmitted. So if the light is transmitted completely, we'll obtain peak something like this. And if it is absorbed at a particular frequency, here we are especially studying the frequency of CH bonds, because we need to detect the ribose sugars present in the sample. So if we observe some activity like this inverted peak, it basically means that our sample has the CH bonds, and in turn it means that there is the ribose is adsorbed. So we'll go to the data one by one. Yeah. First, what they did was they took two samples. One was ribose and kaolinite only. The other was ribose and kaolinite adsorbed with CO2 plus. And as you can see, what they observed was there was no peak obtained in the ribose kaolinite only mixture, but a significant inverted peak was seen in the ribose CO2 plus kaolinite mixture. So we can say that ribose is significantly adsorbed in the presence of CO2 plus system. And in the earlier experiments done by Lambert and team, the failure of that experiments could probably because there was no metal cation used. So that could be a reason that didn't work out. The next thing, since we have established the fact that ribose CO2 plus kaolinite mixtures work well, we can test them at different pH levels. And we observed that significant peak was observed at the pH 7, while there was not much activity in the rest of the pH range. So we can infer that pH 7 is the optimal for adsorption activity of ribose. Next up, since we have to be sure that this process is not very specific, this process is actually specific to ribose, and it's not just about the CO2 plus kaolinite mixture. What we did was we analyzed different kinds of sugar mixtures. So we had different pentoses, arabinose, lixose, ribose, and xylose. And to not much of surprise to the researchers, the activity was only observed in the ribose mixture, of course. So significant peak was obtained in the ribose CO2 plus kaolinite mixture, while no peaks were observed in the rest of the pentoses. So we can say that only ribose shows significant retention activities with the MDC systems as compared to the rest of the pentoses. And then finally, so if concentration of the ribose has some effect, so it turns out that the lowest peak was obtained at the lowest concentration and so on. So the retention factor quite increases with the increase in the ribose concentration.
So now we'll talk about another important technique involved, which is the HPLC. So HPLC stands for high performance liquid chromatography. It is again a chromatographic technique, and the column here mimics the sort of place system that we see that Srushti talked about before. So the column consists of the kaolinite, metal doped kaolinite structures, and how we basically do is the analytes and the sample we want to see are taken into the syringe and injected into the column. There's a solvent reservoir, which is then pumped into the column again, along with the sample to make sure elution happens. And as this is done, the sample travels through the column and all the interactions that we talked about before take place. And as the sample starts coming out to the detector, usually something like a mass spectrometer, detects the species involved and sends the data to a computer, which then gives us our chromatogram. Now the question arises, how do we read our chromatogram? So usually the chromatogram, there's an x-axis and a y-axis involved. So the x-axis talks about the retention time, or what we usually refer to as the retention factor. And the y-axis, it talks about the intensity of the signal involved. So the higher the intensity, the more sample we have obtained. So a quick point to note here before we move on to the next part of the data is that the retention factor is specific to each individual molecule. Retention factor does not change unless the species involved change. What changes is the intensity counts. So the more the sample there is, the higher the intensity counts and the larger the peak will be. If the species changes and the chemical changes, the retention factor changes and the peak will be observed at different points on the x-axis. So that's how you read a chromatogram, and which is why it's a great technique to help isolate and identify species. So these are the data that we obtained, and as you can see, they all are beautiful chromatograms. The first set of experiments was done with a very specific column, the Sharp-Silv UC18 column. That's doesn't really matter what it is. And you see that initially you have like a lot of peaks in the original mixture corresponding to multiple sugars. And when the experiment was done and the supernatant was checked with HPLC, you see that there's a reduction in the ribose peak. The intensity comes down. And after desorption, experiments were done from the Cu kaolinite mixture. We see that there's a significant ribose peak observed. So what this says is that ribose was specifically adsorbed from a given mixture of sugar, and only ribose was adsorbed from the given mixture of sugar. You don't see the peaks corresponding to the other sugars here. This was repeated with another column just to make sure that it was not specifically a column thing. And we see the same results happening again, given a mixture of sugars. And we see that in the original sample, we have all four peaks corresponding to all the sugars. And after absorption experiments, we see that there's a reduction in the intensity of the ribose peak. And when the desorption experiments were conducted, the only signal obtained was the ribose one. And this again confirms that ribose was exclusively adsorbed onto the Cu2 plus kaolinite mixture. And when the solid Cu2 plus kaolinite was taken after the adsorption experiments and checked with IR that Srushti talked before, we see the characteristic peak obtained at 2946 centimeter inverse, and that is the characteristic CH frequency. And we again see that the ribose is retained on the Cu2 plus kaolinite mixture at pH 7. And then when we also talked about initially that you can't really expect that you synthesize ribose and then immediately you get the next step involving nucleoside formation. An important factor involved in this is that you have to stabilize ribose for a long period of time. just so that, when the nucleoside reaction starts happening, you have ribose that is reactive and available. And as mentioned before, unless the chemical species changes, the retention factor does not change. It remains the same. So the experiment conducted here involved basically adsorbing ribose onto the kaolinite clay and then waiting for a couple of days before doing the desorption experiments. And as you can see, the even after around 42 days, you've got like a significant ribose peak. And this shows that the MDC, the metal dope clays, could absorb and then stabilize the ribose over a long period of time, so that whenever you wanted the nucleoside reactions to start, ribose was available and reactive, and you could obtain it easily.
So this entire procedure was performed with different combinations of clay minerals and metal cations, just to be sure. And each time, this is a representation of all the desorption experiments that they did finally. So in all the combinations, in all the experiments, only ribose showed significant desorption activity, because obviously it was the first one absorbed at the first place. So just to be sure, we had all the information that we had, that we needed, and it was seen that the process is very specific to ribose, and it is not just about the clay mineral and metal cation system.
So one of the mechanisms they suggest usually involves clay layers and clay minerals like we would find on the earlier system. So there are two possibilities that the researchers suggest could have taken place. One involves a permeable clay layer and the other involves an impermeable clay layer. So what a permeable clay layer means is that the sugars can travel further down into the system and therefore go further in and not be available to react at the solution. The other possibility is that the clay layer is impermeable. The sugar cannot travel deep inside the layer and is, at the best case, adsorbed onto the surface of the layer. And what the researchers say is needed in this situation is that ribose needs to stay on the top layer. They cannot go too further down, because then it becomes unavailable for the reactions ahead. So in case of a permeable clay layer, what the researchers suggest is that every sugar involved has a different permeability thanks to its different chemical properties. And the other sugars might have permeated way further down so that it's not available to react. And ribose stays at the surface and therefore is easily available relatively to react with the next step and form nucleosides. In case of an impermeable clay layer, we would probably see the mechanism that we've talked about before, where ribose is the one specifically adsorbed and the others are not, and it's just washed away. And therefore, ribose becomes available to react directly. And this is what the researchers suggest could have happened on the earlier.
So after all these experiments, we can firmly conclude that ribose can be enriched in a dynamic separation process based on its unique ligand exchange capability, regardless of its synthetic route, yield, and selectivity, or the sugar formation reactions that we talked about earlier. And we can also say that the transition metal cations were likely to serve as the stationary phase for the isolation of ribose from the sugar mixture on the primordial earth. So this study can also provide a plausible and general solution to the long-standing mysteries of the natural selection of ribose and the related prebiotic synthesis towards ribose. We can also talk a little bit about the scope and impact of this research. So the scope, we will never know 100%, but we can always make speculations and look for a better understanding, just like evolution. Then we can also, through this, maybe answer questions like why other nucleic acids aren't present, for example, the arabinonucleic acid. And the future next step to this research could also be finding mechanisms and finding possible evidences in nature. So yeah, we had some questions, which Sarah will just list.
So while reading the paper, we had our own questions as well. So we see that ribose is exclusively seen in RNA and DNA. We were always curious.
Yeah. So
if even remotely there were other saccharides that do form nucleic acid. I think the only one I could find was arabinonucleic acid, and that was synthesized in the lab. We were curious whether it happened in nature, because chemically we don't see why not. The other question we had while we were reading it is, like nucleic acids, proteins have a directionality involved, like we have D proteins and the L proteins. So we see most of the biological systems have L proteins. We were curious as to why D proteins do not occur in nature, or was there any such selective reactions that took place, or selective situations that took place, like we saw with ribose, that kind of specifically targeted L and not D, and why that happened. We were also curious as to what is unique in the structure of ribose and the chemical reactivity of ribose that preferentially makes preferentially results in it being absorbed onto the MTCs, and why not the other sugars. And we were also wondering how understanding this will also help us understand the bigger picture of the abiogenesis theory and how life originated on the planet. So yeah, that it was a very fun times talking about these questions and what we were wondering during it.
Yes, and before we move forward to the discussion, I'll quickly have some acknowledgements. First of all, thank you so much, Anand sir, for guiding us throughout this journey and giving us this opportunity. I'd also like to thank Abha, who helped a lot in this presentation. It wouldn't have been possible without her. And all my fellow Samyojana peers for all their feedback, who helped us in the presentation. And most importantly, Dr. Rajamani, Dr. Krishnamurthy, and the audience for the patient listening. So these are the few questions that we need to address. First of all, what I liked most about the paper was that initially I wasn't convinced at all with the theory, because it was so systematic and so straightforward. I felt like if so much research is being carried on from years, how can they do it at one go? But the thing is that they did it for years and they finally came up with the solution. So later on, the most important thing that I liked about the paper became the very fact that it is very, very systematic and very straightforward. As a team, I think we didn't face a lot of challenges as such, because we have been friends for long and we have a nice rapport. But because it was a new experience presenting a paper together and on something that is on the interface of our interests. So Sarah is a chemistry student and I'm a biology student. So working at the interface was really fun. We definitely face challenges in terms that initially I was completely perplexed by the paper, but this field is such that even though you understand that even if you give a lifetime, you can't understand the whole field completely, you still want more. It is such an addictive field. So I think I completely enjoyed the whole experience. And Sarah, you could go for the questions.
So if you're open
so
to, go ahead.
I would also like to add that although while I found quite a lot of the techniques involved, like we have to study it on our day-to-day classes and stuff like that. But when Srishti asked me a lot of questions involving the very specifics of these techniques involved and why they were doing it, is when I started thinking about the details involved and seriously giving it some thought beyond the just, oh, I have to study it. So that was my the best part about this for me. So that's going to be my takeaway.
Thank you very much. So we do have a decent amount of time for discussion. So I would initially request our guests, Dr. Krishnamurthy and Dr. Rajmani, to make some comments, maybe ask them questions, and maybe give some advice on how they can move forward.
I would
Dr. Krishnamurthy.
I would like to ask Dr. Rajamani to start, and I will follow up. Sudha, you're muted.
No, I'm saying, no, you should start because you're the leader, I'm the wannabe, so you go first, and then I will deal with the leftover business.
No, a good leader follows, so I will follow you.
Can you go back to the presentation, please? Srishti, will that be possible if you can start the presentation again?
Yes, ma'am. I hope you can see the screen again.
Yeah. So, yeah, I think I will let Ram deal with the set of questions on the discussion slides, because between him and his mentor, they are the resident leaders of everything that is on the discussion slide here. I'm going to go back to a few more specific sort of questions, but prior to that, really good job, Srishti and Sarah. I'm glad you picked this paper. Mainly because I'm biased. I love astrobiology and I love all things about RNA world, and I'm glad in that sense. Having said that, coming from where I come from in terms of what we do, there are a lot of things I find problematic with the data of this paper, but that is something I will not get into because this is something you're not as yet trained extensively to pick, but I will highlight a few things. Can you go back to the first slide?
Sure, ma'am.
The
first graph?
The very first slide, which has your faces, the title.
Okay. Sure, ma'am.
Yeah. What I don't see is where exactly this paper was published. Okay, it's a self-pressed journal, but do you know what the name of the journal is?
No, ma'am.
That doesn't matter. Okay, if you go back out of the slideshow mode, you can stay out of the slideshow mode because it's easier to see the slides. Because one of the things that I feel was problematic was your introduction was all great. Can you keep coming down? Let's go beyond slide 6. Yeah. Keep coming. Yeah. Keep coming. Yeah. 1 second. 1 second. Yeah. Slide 16, please. Yeah. Sarah, I think you wanted to say coordinate covalent bonds, not coordinative bonds. Yeah. There's nothing called coordinative bonds unless I'm wrong about this. Yeah. It's
I wanted to say coordinate covalent bonds. Yeah. But slip of my tongue.
Sounds good. Can you now go to, slide 21, please? I'm just curious, the Shodex KS801 resin.
Yes.
First of all, I have a bit of a problem understanding the y-axis. But having said that, to me, arabinose, xylose, and ribose, they look very close to each other in terms of their preference for that sodium-doped MDS, I think, which is part of that column. So I was wondering if you had any specific comments to make for that matter. Even in the Carbopac MA1, it is not tremendously, okay, maybe a bit different, but yeah. Oh, it was published in chemistry. I see. The Shodex KS801, I was just wondering if you had some more details about the nature of the matrix in it, and if that, because your Shodex SC1011 seemed to have a better preference for ribose, just like your SP0810 Shodex, where you have lead as the exchanging metal, as the metal that is doped. So I was just wondering, how much of this interaction is completely going to be decided by what the ionic status of the metal is?
Yes, ma'am. So our knowledge completely is limited to this paper, and there hasn't been specified what all these columns, like anything about the columns isn't specified. What they did say only was that they performed the modern sugar separation techniques, and that they found that there was some significant retention activity going on. So that's why they moved forward to making it relevant to the prebiotic era. What they did say was that these modern separation technologies were not dedicatedly designed to make ribose the most strongly retained sugar, and so the data should be unbiased to imply that ribose might have the unique ion and ligand exchange capabilities. But I think that is where our knowledge ends, and we'll definitely try to find out more about these columns individually.
More than the column, I think my bigger interest is, and this is probably specifically for Sarah, given her chemistry background. Sarah, any notions about why divalent cations seem to be doing a better job than, say, the monovalent cation here? But I also see the trivalent is doing even better than the other two. So any notions about what this...
If I had to hazard a guess, I would probably say it might have to do something with like the HSAB principle, hard soft acid basis, which say that as the charge on the metal cation increases, the hardness also increases. And since as far as I understand, the coordinate covalent bond is formed with the oxygen, which is also a hard species. Maybe that is a factor involved. But then I also understand that as the metal size increases, that changes things. But perhaps the charge has something to do with it. The higher the charge, the more strongly it can hold on to the ribose, and the stronger the coordinate bond form is. Not to mention, as the size increases, you've got more MTD orbitals, and all that comes into play. So I'm not sure, but if I had to hazard a guess, I'd probably suggest these as an option to look into.
Okay, I have just about a couple of more comments. Can you go down, please? Keep going down and let me look at the lights so I remind myself a tad slow. Yeah. If you come to 28, yeah?
Yes, ma'am.
Yeah. Yeah, I really am not an IR person, and this is where Ram also will have to help me out. I'm not ultra convinced with the IR data. If this kind of a data generally flies amongst the community members, then it's fine. But the peak that you're referring to at 2940 inverse centimeter, I'm not even sure if it's a peak. So I do not know how strong a case you can make about this, because in the B panel, which is now not highlighted, I also find that it was not B, it was the next one. Go to the next slide, 28, please. 28, yeah, yeah. If you really sort of, I feel like even saying that lysose and xylose also stand a chance to bind, because I really am not seeing anything ultra prominent. So I am a little concerned about interpreting, making very strong statements based on this IR data for sure. And again, can you come down? As in, can you roll down the slides, please? Keep coming.
Yeah, sure.
Yeah, that'll be nice. Stop. Yeah, slide 32. Yeah, so this is how they have represented the data in the paper, right?
Yes.
Okay. So what is problematic again to me here is, I mean, I would want a little more details about what the other peaks are which have not been labeled. If you look at panel A, where they're claiming that after desorption from the copper kaolinite, they see only ribose, right? So yeah. I can see how you can't see, I can still say there's a bit of L there because there's a bit of a small bump before the R, but what is that peak after R, which is super prominent and almost equivalent to R, right?
Yeah.
Close to, pretty equivalent to R. And in the next one, in the next panel B, one, I have a problem when they don't tell me what's on the y-axis. That's a bit of an issue for me. On the in the B panel, I don't think the desorption from copper kaolinite necessarily says anything much, because they all look like squiggles to me. If R is a little prominent, then that other peak at 6.25 is also a little prominent. So what is that is the question.
Ma'am, I don't know for sure. Of course, it is too prominent to be called an impurity, but I think they have said that it is.
In some of the slides, they called it an impurity. I think this is just me being critical about what what the authors are saying, which you're trying to communicate, and this is something that you need to imbibe in your literature review process, right? You need to be very critical about these things when you're evaluating them.
Absolutely.
So I think the last question is pertaining to the Sharpsil UC18 column data. Sharpsil, no. Go upar jao 1 minute, sorry. Yeah. 32, not even 32, 31. This is where you introduce, where do you say Sharpsil C18 column specifically?
This one. This is
yes,
the one.
here.
Yeah.
Yeah. Yeah, again, similar concerns as what I raised in the previous data set now. There is still the peak around 19 minutes, which is very prominent. No comments about that by the authors. And I still see lysose binding to ribose, binding to the MDC. So yeah, I'm not fully ultra convinced again with the HPLC data based on which they're making their, at least the HPLC is a little more, maybe thoda zyada points dungi main uske liye, but IR data, I'm really not too sure at all. But overall, good job with you folks communicating this paper to all of us and giving the listeners today a sense of how very broadly the field thinks about these sugars in general and why ribose is the rock star, possibly the rock star pentose, and yeah, giving sort of a very nice broad overview about that in addition to this study. Good job, and I will let Ram take over.
Thank you so much, ma'am, for the different perspective. We really look forward to go out of the paper and read more literature.
Yeah, I will ask you to start with all of Ram's papers.
Definitely.
Yeah.
So, see, this is why I let Sudha go. She did a marvelous job of taking the whole thing apart, in details. And, she really told you how to read a paper critically. One of the things that I see students do when they present a paper is they present the paper and they try to say what the authors are saying, but they don't critically analyze it themselves. And that's what you need to do, which you did a little bit, during your presentations. You expressed certain doubts about how you didn't understand certain things, but, Sudha really laid it out as to how you must ask such critical questions, look at their data. And one of the first and foremost things a scientist does is be skeptical of any claims. And, because that is how you really, get to understand a paper, the claims that they make, the data. You have to read that very closely, as she, as Sudha marvelously pointed out the problems. And, especially with the IR data, I completely concur with her, reservations there. Now, apart from, the paper in terms of, I know this author, and, they wrote to me wanting me to read the paper after they published, and I can, read what I wrote to them. Zhao was the first author, the author, Zhao Wang. He said, he wrote to me saying that, we published this paper, please give your opinion. I said, dear Zhao, it's extremely kind of you to inform me about your paper, which I've already seen. It is interesting work, and as Eshin Moser said, it is important to collect all chemical facts to understand the chemical roots of origins of biomolecules. This approach must be balanced, and that's the important thing that, what I'm trying to convey here is, just because you selected ribose at this level means nothing, and I must say that in all sincerity, means nothing for the emergence of RNA. Okay, because if you look at the selection of RNA, Why is RNA selected? RNA is selected because of a function. No molecule is selected in biology just because it's there. Every molecule is selected within biology because there is a function. The only molecules they are selected that are there if they are building blocks like CO2 or oxygen or methane. But once you are within biology and a sophisticated molecule like RNA, you are there because you function. If you don't function, life does not select you, life does not need you. And remember, RNA is not just ribose. RNA is made up of the phosphate connection and the bases. Without that, the ribose is totally useless. And if you go and look in biology, how ribose is made, ribose is not made by the Formose reaction nor as ribose itself. It's made from ribulose and xylulose phosphate. There are pentulose sugars and always handled as a phosphate. It is also broken down from glucose phosphate. So at this level, the finding that ribose is selectively absorbed on a mineral is interesting at best. That's all. So I go ahead and told him, this approach must be balanced with the realization that the selection of these biomolecules is based on a function that's only possible at a much higher level, where the function can be expressed such as in an oligomer. And therefore, one must be cautious about over-interpreting the results for what it means for origins of life. It doesn't downplay the significant fact that they absorbed, ribose selectively, but to immediately jump and say, this is why ribose is selected in life, is you are not only fooling the public, but you're also fooling yourself. And then I give him, some references from Eschenmoser's work. And Eschenmoser has published quite a lot as to why ribose is unique by studying the different sugars. And you raised a very good point in your slides, saying that why there are not other sugars in an RNA, why it is ribose. That is a very good question. And that's exactly what Eschenmoser has answered with his work. Why don't you see other saccharides? So he made hexose nucleic acids. He made glucose nucleic acid. He made arabinose nucleic acids. All of them do not base pair. And so you can argue, ribose is selected because it base pairs. But base pairs at what level? At ribose? Ribose has no base pairing. It base pairs only as an RNA. So you can't just, it's like taking, just only the nose portion of a baby and saying, that nose exactly looks like the father. And you construct a complete human being. I'm just reminded, have you people seen this famous photograph of a face on the Mars? There is a very famous figure, and it looks exactly like a human face on Mars. And people immediately said it must be intelligent life that made it. Okay, that is the type of connection anybody would make. Be it just an amino acid in a meteorite, or an amino acid in a Miller-Urey experiment, or a ribose in a Formose reaction, or adenine from the very famous Oro reaction coming from HCl, and immediately claiming to be building blocks of life. And ask yourself this question. Let us say you do make ribose. Nobody has really shown that ribose reacts with the canonical nucleobases to make a nucleoside. Okay? So that has never happened. It's very difficult to do that. And Thomas Carroll has worked very nicely, and he had shown, these kind of alternative bases that reacts with ribose. Sutherland has started from the oxazolidone approach. So there are various ways people have come about it, but nothing really answers the question really as to how. And ask yourself another more important question. These authors started with pure sugars, right? They bought these pure sugars. Imagine if they had, and I'm sure they did try that experiment where they took the Formose reaction itself and see whether they can select ribose out of the messy reaction. And I'm sure they failed, but they don't say that. Okay? And there is a reason why authors do this, because they need to get good papers for their students so that they can go ahead and get a job or a postdoc or a good graduate student or a good PhD. You don't get a PhD with messy chemistry anymore. Okay, gone are those days, and you need to really look out for the students' future as well. You can't just put them on such messy projects. And one of the things that Eschenmoser taught me, which I really, if you want to be in this field as a PI and make your life in this field of origins of life, he said, whatever you do, select a project by which, if you take away the importance of prebiotic chemistry, it still stands on its own as a contribution to chemistry, biochemistry, biology, or something else. Because if you don't, then it's not an interesting paper anymore. So, but it was an excellent job that you both did in terms of presenting the paper, and you did quite good justice to the the authors. If the authors were here, they would have been proud to see their work presented this way. Really, you went through the paper in very great detail. But at the same time, as Sudha pointed out, I cannot stress this enough, and I second it again and again. You need to be more critical in reading the paper and bring up your own questions about the data itself if you have doubts, about their own conclusions based on the data. And some of the things that Sudha pointed out, ask that question, what is that extra peak? Why didn't they identify that? If they don't, then you should put that up, in your own slide there, saying that they have not identified this peak. That would be very important. That's a good part of growing up as a scientist, personally as a scientist, to question the paper. Because if you are able to find the answer after you ask the question, then it's more convincing than just believing what the authors claim. Okay, so bottom line, nice work. This might be a good way, but there are a lot of issues that I have with the paper in terms of prebiotic claims as well. For example, they are using copper +2. Copper +2, if you go to the anoxic conditions where there is no or very little oxygen, it should be Cu+1, just like it should be Fe+2 and not Fe+3. So where did they get their copper +2 from if they wanted it in a prebiotic conditions? Those kind of questions will come up. They need to justify that. I'm sure that they might have justified it in the paper, but then there are all these ifs and buts that go along with that, which makes it a little hard to connect to prebiotic chemistry importance of the paper, but it does not detract from the fact that they did select ribose. So those two are two different things. Okay, I'll stop here.
Thank you so much, sir and ma'am, for giving us this perspective. I would like to mention that in the beginning of the paper, we had a whole lot of questions and we were skeptical about a lot of things. But as students, what we forget is that we are allowed to ask those questions. What we think is that this could be because of our lack of knowledge and we need to go through a lot of literature in order to match and be able to analyze a paper. But thank you so much for reminding that we can actually critically analyze every paper that we are given to read and also ask questions regarding it. So thank you so much, sir and ma'am, for that perspective.
And this is exactly what these presentations are there for. We also learn because we may not have had the time to go through that paper in as great detail as you presented. So it was a good way for us to learn about the paper as well.
Thank you, sir.
Thank you so much. This was like a model session for us to kind of mimic and promote this. So we actually now have a benchmark. In Samyojana, we have a new benchmark so that we can use that as a reference for our future deliberations and discussions. So I would love to have, I do know you have other commitments, but I would love to see you guys be there for at least 2 more talks if possible, but I can understand if there are time pressures. So I will invite Kiravani, Medha and Nidhi to show talk about the paper that they've chosen.
So I'll have to now sign off. It's because it's close to 12 a.m. for me. So see you all then. Thanks again for this opportunity.
Sir.
Yeah, very nice to see you, Sudha. Yeah.
Same here. Bye. Ciao.
Kiravani, Medha and Nidhi.
I'll just share my screen. I hope everyone is able to see the screen.
Yes, please go ahead.
Okay. Yeah, so good afternoon, everyone. And firstly, thank you so much for the opportunity to present here today. My name is Kirvani, and I'm an integrated MSc student majoring in systems biology from the University of Hyderabad. I'm joined by my members of my team. We have Nidhi, who is a BSc biotechnology student. She's from Nagpur, and then there is Medha, who is a BSc Pure Bio student from St. Anne's College for Women in Hyderabad. So our paper is titled Comprehensive Molecular Profiling Identifies FOXM1 as a key transcription factor for meningioma proliferation. So without any further ado, we'll get started. So to give a brief introduction about what our paper is going to be, firstly, we are going to use the word meningioma extremely often from now. So I'd like to introduce what we are going to talk about. So meningioma is the cancer of the meninges. So meninges are the coverings of your brain and the spinal cord. They're the layers that cover your brain and the spinal cord. And the cancer of those meninges is what a meningioma is. And it is one of the most common primary CNS tumor. Then surgery and radiation. So basically, if a person gets a meningioma, the most common treatment for that person is surgery and radiation. And they are like the mainstays of meningioma treatment. But these are insufficient to control high-grade meningiomas. So meningiomas are, we'll get to that a little later, but like meningioma, any tumor basically is classified into a benign or malignant, or like it's staged basically, stage 1, stage 2, etc. So the higher the stage, the higher the malignancy, and it's the higher grade. So when the tumor progresses to a higher grade, surgery and radiation are insufficient to treat those tumors. As a result, there are no curative therapies or effective molecular treatments to treat these aggressive or high-grade meningiomas. Therefore, it's imperative and it's important that we understand meningioma biology to identify the biomarkers that can help us, develop novel therapies to improve the outcomes, because there's no proper treatment, and hence it's important that we know the molecular mechanism, or like how exactly does this tumor progress or anything. So a biomarker is like any biological molecule that's a sign of a normal or an abnormal process, which actually helps us indicate a disease. So yeah, then like I was saying before, staging, like the WHO or the World Health Organization categorizes meningiomas into grade 1, grade 2, and grade 3. Grade 1 is the normal benign meningioma, grade 2 is an atypical meningioma, and grade 3 is anaplastic. So as you progress from grade 1 to grade 3, the difficulty in resecting or getting a treatment, or everything gets complicated. So this treatment, this categorization is based on the mitotic activity and the histopathologic features. Mitotic activity is like how fast or how aggressive the cells divide. Then histopathologic features is how the cells, these cancerous cells, look under the microscope. This is also like the you use the histopathologic features to identify which tumor it is. It's basically the biopsy, as we call it in the like normal conventional culture. And so for clinically aggressive meningiomas are so the issue is that the outcomes are worst for this WHO grade 3 or the anaplastic meningiomas. So what happens is, like by now we've understood that higher the grade, higher the aggressiveness, or worse the output. But what happens is, even in lower meningiomas, like a subset of low-grade meningiomas, are also prone to recurrence and poor survival. Like even though they are like lower, or they have a reassuring histopathology, they are prone to recurrence and difficulty. So when we use the word clinically aggressive meningioma, it's not necessary that it's only a grade 3 one. It can also be a lower one. So any meningioma that is a high grade or has a local recurrence or both, that's a clinically aggressive meningioma. And our paper today is completely about these clinically aggressive meningiomas. Now, who gets a meningioma? Like what exactly happens with a meningioma? So usually there's a genetic condition called neurofibromatosis type 2. So the patients who have this genetic condition, but before that, neurofibromatosis type 2 is like, it's a condition in which tumors grow in the nervous system. Like the cells have a division, like the division in the cells goes haphazard, and they have frequent growth of tumors. So the patients with that disorder have an elevated risk of developing meningiomas of all grades, like even benign or stage 1, stage 2, anything, patients are at more risk to getting a meningioma. And this is due to the inactivating mutation in the NF2 gene. So, and on top of that, when you sequence the NF2 gene, the whole genome or whole exome sequencing, they identify that this AKT1, KLF4, TRAF7, and PLR2A, these genes are responsible for this NF2 mutation or causing meningioma. But beyond the NF2 mutations, even the people without an NF2 mutation have a recurrent meningioma. This is rare, but there are cases of meningioma being reported in them as well. And their genomic techniques are not identified key, like which mutation is causing this, or why exactly is this patient getting a meningioma. So recent work has focused on identifying it has focused on using multi-platform characterization techniques using the genomic, epigenomic, and gene expression techniques to identify the meningioma biology, like to identify the biomarkers or to identify the molecular mechanisms underlying this whole, the progression of meningioma, or like everything about a meningioma. But the point is, despite all of these advances, despite all of these novel targets and developments, it's difficult. It's like the outcomes of meningioma remain elusive, and the molecular drivers of aggressive meningiomas are incompletely understood. So our paper is something that talks about this. And the picture over here, it talks about the different types of meningiomas, like types with not with reference to staging, but like different locations where you can get a meningioma, and different locations of the brain. So there's like different types of meningioma, and this is a picture representing that. Then, so now I'd like to give a brief summary of what we're going to talk about in this paper. Like now that we have an idea of what a meningioma is or what's going to happen in this paper, I'd like to talk about what is discussed in this paper. So our paper is published in Cell Reports, and yeah, so what happened is that in this paper, they took 280 human meningioma samples, and they performed the different multi-platform methods such as RNA sequencing, immunohistochemistry, whole exome sequencing, DNA methylation arrays, and targeted gene expression profiling. So we'll talk about each of these methods as we move further. And then after that, they try to define the molecular profile of an aggressive meningioma. In the transcriptomic analysis, identify FOXM1 as the key transcription factor for meningioma proliferation, as the title suggests. And this is one of the markers for poor clinical outcomes in aggressive meningiomas. In addition to this, we also discovered the genomic and epigenomic factors that are associated with this FOXM1 activation. And finally, we define, like the paper defines FOXM1 and WNT signaling axis that is associated with this gene expression program. Why, like the whole pathway of why is it having poor clinical outcomes, or the proliferation, why is it showing that high proliferation in this meningioma cells. And like to summarize all of this, we find the multiple molecular mechanisms that converge on a POX7 axis in an aggressive meningioma. Now, this is the graphical abstract of the paper. And so what happens is that using this multi-platform molecular profiling, we identify the WNJ signaling axis, which is associated with the cell proliferation and the marker of clinical outcomes. Then so basically, to summarize everything that I said, this paper helps understand the mechanism of meningioma and why is it having poor clinical outcomes. Now, how this happened, like now we'll get into the details of the paper. So to elucidate the molecular drivers of meningiomas, first they developed an integrated database, getting data from 261 patients who underwent surgical dissection of 280 meningiomas. Dissection is like basically removing the tumor. So there are 261 patients who underwent surgery to get their meningiomas removed, and they got like around 280 samples. Now they performed the RNA sequencing, immunohistochemistry, whole exome sequencing, DNA methylation profiling, and targeted gene expression profiling techniques. So now my friend Nidhi will talk about a couple of these techniques.
Yes.
Over to you, Nidhi.
So, yeah, to elucidate the molecular drivers of meningioma, as Kirwani said, they performed several techniques. The techniques were RNA sequencing, immunohistochemistry, whole exome sequencing, DNA methylation profiling, and targeted gene expression profiling. Let us go through RNA sequencing in detail. There are many classes of RNA molecules. mRNA carries genetic information from DNA to the cellular machinery that synthesizes proteins. Suppose we have two groups of cell, one which is a normal cell, one group of is of normal cell, and the other group is of mutated cell. With the help of RNA sequencing, we can measure the gene expression in normal cells and then use it to measure gene expression in mutated cell. Then we can compare the both and figure out what is the difference between normal and mutated cells with the use of RNA sequencing. For example, in a normal cell and a mutated cell, gene 1 has no difference and the gene 2 has big difference. The gene 3 has a slight difference. So with the help of such comparisons and RNA sequencing, we can find out the differences. To perform RNA sequencing, there are certain steps to be followed. The first is isolation of RNA. After that, break down the RNA into small fragments and convert the RNA fragments into double-stranded DNA, because as a double-stranded DNA is more stable and can be easily amplified and modified. This double-stranded DNA is also called as cDNA, complementary DNA. Then adding DNA sequence adapters. After addition of sequence adapters, the DNA is called as A-DNA. Adapters allows the sequencing machine to recognize the fragments. It also allows to sequence different samples at the same time. After that, PCR amplification is done, and after quality control. Quality control is done to verify library concentration and verify library fragment lengths. After this comes RNA sequence library. After the whole process, the RNA sequence library is obtained, and that can be read the sequencing and can do the interpretation. Mainly, RNA sequence analyzes the transcriptome, indicating which of the genes encoded in our DNA are turned off or turned on, and to what extent. And understanding transcriptome is key if we are to connect the information in our genome with its functional protein expression. This allows scientists to understand the biology of cell more deeply and assess changes that may indicate the disease. The second technique is immunohistochemistry. It is a laboratory method that uses antibodies to check for certain antigens or markers in a sample of tissue. The antibodies which are used are usually linked to an enzyme or fluorescent dye. After the antibodies bind to the antigen in the tissue sample, the enzyme or dye is activated and the antigen then can be seen under a microscope. IHC, that is immunohistochemistry, is used to diagnose diseases such as cancer. It may also be used to tell the differences between different types of cancer. The next three techniques will be explained by Kirwani.
Thank you, Nidhi. So these two are the major techniques that they have performed. So I'll give a brief explanation of the other three techniques. So whole exome sequencing, like as the name suggests, it's a form of DNA sequencing where only the exons or the part of DNA that codes for the polypeptide is sequenced. Then basically the protein coding regions. Then there is DNA methylation profiling. So DNA methylation refers to the adding of methyl ends in the DNA. It is essential for silencing these retroviral elements that regulate tissue-specific gene expression, and it's basically used in identifying the X chromosome inactivation. And so they try to use this DNA methylation profiling to look at the genes in this meningioma tissue, and like the tissue specific gene expression, the meningioma specific genes ka expression. Then there is the targeted gene expression profiling. So this provides a convenient and effective method to identify the pathway of the genes that we are interested in, like the complete picture of the gene that we want to look at. So that this is a brief explanation of the techniques that they performed. Now going back, So after performing all of these techniques, they try to characterize the transcriptomic, genomic, and epigenomic features of meningioma in relation to their clinical parameters. And they found, they discovered two distinct transcriptomic clusters of meningiomas. And they showed that the aggressive tumors from each cluster, like from each type, these aggressive tumors had an elevated Fockhead box-M1, which is the full form of FOXM1 mRNA expression. So they discovered this FOXM1 mRNA, and its expression was high in each of these transcriptomic clusters. So what exactly is FOXM1? So FOXM1 is a pro-mitotic transcription factor. This is required for cell proliferation during development. But what happens is, in cancers, not only just in meningiomas, like for example, in hepatocellular carcinoma, which is a cancer of the liver, or in prostate cancer, or in glioma. Glioma is another type of brain tumors. The FOXM1 has higher rates of proliferation in these cancers also. And it is shown that FOXM1 expression has been highly expressed in these cancers tumors in compared to the normal tissue. And it's particularly enriched in invasive tumors. Invasive tumors are like the ones that recur like continuously as in a short period of time. And they found that the FOXM1 protein expression is associated in meningiomas. It is associated with increased cell proliferation and poor clinical outcomes. Moreover, they also found that meningiomas were characterized with, these type of meningiomas were characterized with high somatic mutation and a genome-wide hypermethylation of genes. So these basically inhibit FOXM1 activity, but in these tumors, they do not inhibit FOXM1 activity, as a result of which this FOXM1 expression is like really high, which leads to an increased cell proliferation and poor clinical outcomes. So after identifying this FOXM1 expression and like this FOXM1 mRNA, they defined a FOXM1 WNT signaling axis. So this was associated with the mitotic gene expression program, like with poor clinical outcomes. So basically, after identifying this protein, they took the whole pathway of this whole signaling axis with how to drive, like what exactly drives this meningioma proliferation and tumor growth. And so yeah, that's exactly what's given in the picture as well, with the what happens with the normal tissue and what happens with the dissected frozen tissues. So now my friend Meda will go on with the rest of the paper.
Thank you, Kirvani. As my friend explained about how the proliferation of meningiomas occurs, and also that FOXMI protein is also showing a related.
Ida, your voice is not clear. Can you be a little louder, please?
Yeah, sure, ma'am. Can you hear me now?
It's very feeble.
Okay.
Try again, say something. Otherwise, it's hard to know whether you're.
Oh, can you hear me now?
Yeah, much better. Yes.
Okay, ma'am, sure. As my teammate carry on with how the FOXMI is proliferating on the meningioma tumors, and also that the FOXMI, the tumors are really much more in the somatic tissues, but it shows a higher reform and also a higher rate of attack in the tumors like meningiomas. Thus, from many clinical perspectives that we have found out that it's not in the differences of the patient, the sex, or the meningiomas location, but not even the age. But sometimes these parameters are due to the somatic disturbances or the genetic differences that is due to the diseases or the disorders of an individual person. And this is also due to the metabolic rate. The metabolic rate is shown high in the male sex, and the higher grade of meningiomas are more. And this is also due to because of the phosphorylation of the genes. And that's where they reveal the tumor location and the histology subtype by the gene ontology techniques. And by these analyses, it could able to reveal the cell division and the mitosis genes were higher and goes with the transcripts in the WHO grade 3 meningiomas when compared to the grade 1 of meningiomas. And it is also found out that the FOXMI protein, when it is sequenced along with the chromatin immunoperception sequences, it showed that grade 3 WHO meningiomas were with a higher attack and in a higher diffuse in the parts of a brain when compared to the grade 1 of WHO. So this is due to the histopathological disorders and also the mitotic activity of those meningiomas, which is supported by the FOXMI expression. Actually, the FOXMI expression were taken to show how the meningiomas work so that we can find the location, and also to control the meningiomas that are being growing in the meninges. But it shows that FOXMI not only tried acting as a foe, but it has been a friend by giving a more support to the meningiomas to adapt, grow, and proliferate in the surrounding regions of meningiomas. And this is one of the reasons there's a poor clinical techniques, and it shows the poor survival rate in few of the patients. And also it's also, so this how we summarize is that the cell division is higher in this regard, and clinical techniques do not show much result in the meningiomas and other techniques. So what we kind of felt is these techniques were not easy, or it wasn't really favorable at this point of time. Now I would like to hand over this to my friend Nidhi.
Thank you, Megha. So, moving further, they said that high-grade meningioma are characterized by elevated cell proliferation. Cell proliferation is the process by which a cell grows and divides to produce two daughter cells. Cell proliferation leads to an exponential increase in a cell number and is therefore a rapid mechanism of tissue growth. In multicellular organisms, cell proliferation is tightly controlled by gene regulatory networks encoded in the genome and executed mainly by transcription factors. Uncontrolled cell proliferation can be the cause of the cancer. To determine if FOXM1 was associated with meningioma proliferation, they quantified FOXM1 protein in a total of 52 tumors constituting both indolent and aggressive meningioma. Now let us quickly go through indolent meningioma. Indolent meningiomas are grade 1. This means that the tumor cells appears indolent, which is slow growing under a microscope, and the tumor is not spreading very rapidly. And talking about aggressive meningioma, which is very difficult to treat, especially when they are surrounded by nerves. They grows very rapidly. After that, immunohistochemistry for FOXM1 revealed significant intrameningioma heterogeneity that was restricted to meningioma cells, as denoted by co-staining for SSTR2A. Somatostatin receptor is widely expressed in meningioma. SSTR2 is extensively studied as a marker and target for the diagnosis and treatment of meningioma. Co-staining for the cell proliferation marker MIB1 demonstrated a strong association between FOXM1 and mitotic activity and meningioma. MIB1 labeling index is the percentage of immunoreactive tumor cells in the evaluated area. Also, meningioma proliferation, as defined by M1BI marker labeling index, it was associated with decreased LRFS and decreased OS. LRFS stands for local recurrence-free survival. The length of time after primary treatment for cancer ends, then the patient survives without any signs or symptoms of the cancer, is defined as LRFS. And OS, which is overall survival, it is the length of time from either the date of diagnosis or the start of the treatment for disease that patients diagnosed with the disease are still alive. Consistently, they found that high FOXM1 protein expression, as defined by greater than or equal to 75 nuclei per mm square, was also associated with decreased LRFS and a strong trend towards decreased OS. Thus, high FOXM1 protein expression in meningioma is associated with increased cell proliferation and poor clinical outcomes. Hematoxylin is used to illustrate nuclear detail in the cells. The depth of coloration is not only related to amount of DNA, but also the length of time the sample spends in the hematoxylin. So they can say that when there is high M1B1, when there is high M1B1, there is decrease in LFRS and OS, and when there is high FOXO, there is also decrease in LFRS and OS. Further part will be explained by Kirwani.
Sorry, I didn't realize I was on unmute, so really sorry for this. So yeah, thank you, Nidhi. So now I think you got an idea of all the techniques that were done and how they came to identifying this FOXM1 or WNG signaling axis and how it performs cell proliferation and gene expression. So to confirm the associations between this elevated FOXM1 mRNA expression and the FOXM1 target gene expression and poor clinical outcomes, they performed targeted gene expression profiling with a validation cohort of 96 meningiomas. Now, to they identified that this FOXM1 transcriptome protein expression was common in high-grade and recurrent meningiomas. Now, this FOXM1 protein directly interacts with another protein called beta-catenin to transduce WNT signals and regulate cell proliferation. So, what exactly is this WNT axis or WNT pathway? So, WNT or this WNT proteins. So, WNT proteins are involved in regulation of cell cycle, cell differentiation, cell motility, cell polarity, and cell death, like all of these, which are responsible to make sure that this cell does not become cancerous. So this FOXM1 interacts with beta-catenin, and that transduces this WNT signals, and this whole regulates cell proliferation. So this beta-catenin is the central component of the WNT signaling pathway, and it is responsible for regulation of apoptosis and differentiation as well. So to further analyze this relationship between the FOXM1 gene expression and clinical markers of this aggressive meningioma behavior, they found that high FOXM1 expression was the most important factor for tumor recurrence as well, not only just progression, but also for recurrence. This high FOXM1 expression is needed. Now, to determine if the WNT pathway was associated with poorer meningioma outcomes, they assayed the beta-catenin localization using immunohistochemistry techniques, having 232 other meningioma samples, and found abundant cytoplasmic beta-catenin staining in meningioma and very less nuclear staining. So this is visible from the figure 6. And then they put it under confocal microscopy and realized that, and then they identified the, and then they stained, sorry, and then they identified, I'm really sorry. And then they stained the FOXM1 and beta-catenin using immunofluorescence. So that's seen in the figure F, sorry. Now, to elucidate the further the transcriptional network that like the pathway that transduces FOXM1 or this WNT mediated cell proliferation, they selected genes that are enriched in the WHO grade 3 meningioma from the previous RNA sequence analysis, which revealed two distinct clusters like I was talking about in the beginning. And they found that like both of these clusters had FOXM1 as a common target. So one of the clusters had FOXM1 target high, and the other one was a low target one. And they identified that the first cluster had a decreased LRFS, and like the one that Nidhi was talking about earlier, and the other one had a higher LRFS and OS in the clusters with a low target FOXM1. Finally, to visualize the biologic impact of this FOXM1 target gene, like how exactly is this responsible for higher cell proliferation, they constructed a protein-protein interaction network, which is like the figure M. And they identified that FOXM1 is also interacts. Oh, but before that, a protein-protein interaction network describes the physical interactions between proteins, which are taking place to mediate the assembly of proteins into protein complexes, like how these proteins interact, what exactly happens, the whole pathway. So the FOXM1 target protein-protein interaction network could be seen was developed and is the figure M. So this shows that FOXM1 interacts with other proteins such as STOP2A, CCNA2, CKS2, which are also responsible for this cell proliferation, making a hold of the cell proliferation and differentiation. So in summary, the data across all of these multiple platforms, multiple molecular platforms and patient cohorts had identified FOXM1 and WNT signaling axis that promotes cell proliferation, gene expression, in an aggressive meningioma. Now, Meda will go ahead for the studies.
Thank you, Nidhi. Sorry, thank you, Kirvani. So we have seen that the genomic expressions and also the epigenomic expressions are supporting the proliferation of genes because of the FOXMI and the beta catenin, the
Audio issues again. Can't hear you at all.
Hello, am I audible?
Yeah, now you are, but we lost you completely for about 10-15 seconds.
Oh, okay. So due to the genomic and the epigenomic expressions, and also the FOXMI and the beta catenin coordination to end the proliferation of the gene expressions and the hypermutilation that we have taken to help us to end these tumors. But we have seen that, Nidhi, can you please change the slide? Kirtmani.
Yeah.
Yeah, thank you. So we have seen that because of both of them, when it is merged, we can see that this acts as a protagonist more than an antagonist, and by both of the proteins, instead of acting as an against, but it acts as a pro member to help it to merge it more, but also it shows a more somatic burden on the patient. And the meningiomas keep on proliferating, and they do find a new location to make it a much more better place to merge and also to grow it. And there are some times where there's a chemical radiation that is being given to a patient, but even before that we can see that there's a somatic location, and there are somatic cells which do show the location of tumor control, but also there's also a cause or a probability of the tumor growth in very sensitive places. And SRPF1 is a protein gene coding protein, and it also helps in understanding the pathogenesis of tumors in the somatic cells. So we have taken this along with the FOXMI protein so that we could control the meningiomas, but somehow this shows the better growth of meningiomas, and the WHO1 grade is bit independent, is seem to be dependent, whereas WHO grade 2 and grade 3 are independent. And WHO grade 1 shows a bit of inclination towards WHO grade 3. That's by supporting, and also the dependence and growth of tumors in different locations of cranium. So the conclusion where we can say is that we don't, by taking the different proteins like beta catenin in FOXMI and the genomic analysis and SRFPI proteins, we couldn't able to stop this, but this shows a poor clinical outcomes also by the data and the statistical analysis that FOXMI protein isn't a suitable protein expression to end this meningiomas.
Thank you, Meda. So moving further, there are a couple of questions that Sir wanted us to answer. So first was like what each of us liked the most about the article, story or question that we presented. So I really liked how this paper had molecular wet lab techniques, and then making transcriptomic networks and protein-protein interaction, which is like Zylab computational techniques. I really liked how it was an intersection of both wet lab and Zylab techniques, and that's and the fact that they came to a conclusion ki, okay, this FOXM1 is the one that is driving meningiomas. Like they gave a fact, and I really like how they undertook this kind of research. So the other questions will be answered by the other members of my team.
Sorry for the audio issues in the previous slide. Thank you for the opportunity. So what do we like and learn the most as a team during the past weeks in Samyojana? So when we as a team, we learned a lot as a team by having a good time management. Sometimes we couldn't able to make it because of our challenges, like three of us are from different colleges and different places, and our syllabuses and also the timing of classes weren't merging. So it was really a hard time for us to make time and discuss the points and make a huge preparation. It could able to happen in a very short span of time, that I'm really grateful for. And we learned from many other teams that they are really doing good, and through them we took an inspiration to try to present it better and make our content eligible for this seminar. And I, as an individual, I chose this paper because I'm really interested into the molecular sciences of biology, and the proliferation of meningiomas, that is because of the protein which has to be acted as an antagony, but this acts as a protagony. And this is something new that I wanted to know about this paper, and this has been a good thing. And the next questions would be taken by my other teammates.
What lessons you, as an individual and as a team, are taking from this experience in your journey ahead? This was a completely level of professional level presentations. We've never done this before. So definitely I would follow the format, how to present, and I'll take the and learn from the other teams how they performed, and will take this ahead in journey.
Thank you, Kiravani, Nidhi, and Medha. We will have time towards the end, so we'll come back to this. So just to keep ourselves on track, can we move to the will it be okay? Can we do a discussion of your talk towards the end? And we'll go with the next group so that we can end before the lunch session on time, or at least as close to our schedule.
Sure, sir, we can.
Yeah.
Sure, sir, no problem.
Thank you. So Rachna, Manaswini, and Gayatri, are you ready?
Yes, sir.
Yes,
I'm
sir.
sharing my
Yes, sir.
screen.
Take it away. Is it the network delay, Rachna?
Maybe.
It's taking a long time, huh?
Okay, just 1 second. Yeah.
Do you want to stop and yeah? Try stopping it and starting again.
Am I audible?
Yeah.
A very good afternoon to one and all.
I am Rachna Golikar, a final year BSc student from St. Anne's, and my teammates Gayatri and Manaswini. Today we are going to discuss on bacteriostatic antibiotics promote CRISPR-Cas adaptive immunity by enabling increased spacer acquisition. So throughout our presentation, the two important things that we'll be discussing on is about CRISPR-Cas and antibiotics. So let's start.
Yeah, we keep hearing the CRISPR is going on to revolutionize medicines, the way we fight cancer and maybe even create a new humans, and we agree with that. Like most things in molecular biology, it is first identified in E. coli, and if we break apart the acronym, it stands for cluster regular interspace short palindromic repeats. It's huge, but it doesn't tell you the two main parts found in CRISPR. Of all we have repeats this are going to be short segment of DNA, so 20 to 40 letters in the length, and they will be palindromic. Remember, a palindromy is a sequence of letters that reads the same left to right, never odd or even. The reason why that when you transcribe the DNA, you make the RNA that forms a little hairpin strands. So we got the repeats; those are all identical, one the another after another, but they are interspaced. They also identified a number of genes associated with CRISPR. So this CRISPR associated or Cas gene, now Cas gene make a Cas protein. The Cas protein in general are going to be helicase. Those are the proteins that unwind DNA and nucleus, that cuts the DNA. So the idea was perhaps this is an immune system of bacteria, a way they could fight the old nemesis, the bacteriophages, and exactly what's going on. But researcher thought if we could hijack the CRISPR system, we could use it because this is a living cell here to either in active genes or maybe even embedded in new genes. In their view, a CRISPR system is an immune system that was identified in bacteria and then modified in humans. And so what's in the middle? Let's keep this curiosity to know more in the further step.
How is CRISPR-Cas immunity activated against phages? So here in the first step, we can see that the virus is attacking the bacterial cell. After virus attacking the bacterial cell, new spacer is released from the virus and integrated into the CRISPR sequence. And the first step and the second steps are called as adaptation. In the third step, we can see that CRISPR RNA is formed, and this step is called as production of CRISPR RNA. And in the fourth step, we can see that the CRISPR RNA guides molecular machinery to target and destroy viral genome, and this step is called as targeting.
Yeah, antibiotics. Antibiotics plays a major role in our article. So what's antibiotics? Antibiotics are medicines that fights bacterial infection in people and animals. So as we all know, what's our antibiotics, and clearly, and thanks to COVID, it is now using what we know how it works. And here there are some antibiotics: chloramphenicol, tetracycline, erythromycin, trimethoprim, carbamazepine, ceproflexin, streptomycin, gentamicin. And there are four different structure of chloramphenicol, tetracycline, erythromycin, trimethoprim. And here chloramphenicol and erythromycin binds to 50S ribosome of bacteria, and whereas tetracycline and trimethoprim binds to 30S ribosome for bacteria. And here erythromycin has a complex structure which has an exception in the results. Let us see about antibiotic activity. So here researcher took an overnight culture and they performed a serial dilution. They took 1:10 to the power, and they performed twofold serial dilution and plated in 96 well microplate, and they incubated for 18 hours and at 37 degrees, and they observed for growth. By observing growth, they gave minimum inhibitory concentration, the lowest antibiotic concentration with no visible growth. And by this no visible growth, they plated again on the agar plate, and they observed that there is less than 5 colonies, and they gave minimum bactericidal concentration. The lowest antibiotic concentration results in 99.9% decrease in initial inoculum cell density. By this ratio, like MBC by MIC ratio is used to estimate the antibiotic activity. High MBC by MIC ratio more than 1 concentration prevents the growth and does not kills the cell, and they gave as bacteriostatic. And with low MBC and MIC ratio, concentration kills majority of cell, and they gave as bactericidal. Yeah, we came back to the same slide where the first floor are gave as bacteriostatic and the next four as classified as bactericidal.
Now that we got to know the antibiotic activity, that was the main part of the paper, let us start the first important experiments that they did. And the first is bacteriostatic antibiotics promote CRISPR immunity. And let us see what all experiments they did behind proving this result. So in this research study, they have used PA14, that is Pseudomonas aeruginosa, and PA14IF as the bacterial strains, and phage strains DMS3-VIR and DMS3-VIR ACR-IF1. So let us begin with our overall experiment for this result. So they have taken the PA14 cultures along with the phages, DMS3-VIR, and antibiotics, and they have cultured in a rich medium. So individually, they have found out the results. So first, they have checked the activity of antibiotics. So as you can see on the screen, the OD growth curves for all the antibiotics like bacteriostatic and bactericidal. So the black color line talks about the no antibiotic treatment, whereas the red color represents the antibiotic treatment. So except for ciprofloxacin, that is CIP seen on the screen, they formed cell aggregates. That is why those are the fancy lines that is shown. But chloramphenicol and tetracycline had strong effects, and you'll get to know how they showed the stronger effects on the bacterial growth. And antibiotic effect on exponential growth rate measured from OD values. So you can see on the y-axis, there is the relative exponential growth rate versus treatment of antibiotics. So how did they calculate the exponential growth rate? The ratio of exponential growth rate with antibiotic over the average exponential growth rate of no antibiotic control was taken for the calculation, and they inferred that chloramphenicol and tetracycline had strong effect on bacterial exponential growth rate, whereas ERM had little effect on the exponential growth rate, but it slowed growth when bacteria reached higher densities. And most antibiotics delayed phage epidemics and the evolution of phage resistance. So until the previous slide, we got to know the antibiotic effect. Now let us see the phage density or the phage resistance in the bacterial cultures. So this is the overview of both the bactericidal and bacteriostatic antibiotics graph. So one day post-infection, they found that there is increase in the phage density for few of the antibiotics, but also they found that there is no phage resistance. And in this graph, you can see on the y-axis, there is proportion of resistant clones at one day post-infection versus phage density at one day post-infection. So you can see that there is like nil antibiotic resistance on the 0 day, whereas post-day, on the third day of the infection, like the same graph, like proportion of resistant clones versus time, they found that there was increase in the resistance of antibiotics at the third day for all the bacteriostatic antibiotics. And also there is, the same result was found even in the bactericidal. So finally, after knowing the antibiotic activity and the resistance, they performed the evolution experiment. They did this in the glass vials where they have taken the overnight cultures and they have performed dilutions in the absence of phages, including antibiotics. Again, they have added the phages to the fresh medium. Again, they have monitored the cell densities and phage titers for 3 days, and then they enumerated the colonies on agar and plaques on PA14 cells. So this helped them to identify the phage resistant type. That is the important part of our first result. So to know that they have cross-streaked their cultures on the agar medium on DMS3 VIR and DMS3 VIR ACR IF1 phages. So basically cross-streaking is done for the to know the antibiotic activity of the bacteria. So there are two resistant types that they found, that is SM clones. It means that surface modification clones which were resistant to both the phages, that is smooth, they were found as smooth colonies. And CRISPR-Cas immunity was resistant to DMS3 VIR but sensitive to DMS3 VIR ACR IF1. And now we know the resistance, what is the resistance in the experiment. So their aim was to understand how antibiotics shape the population and evolutionary dynamics of P. aeruginosa during phage infection. So we can see on the graph that there are sensitive CRISPR-Cas and surface modification in 3 different colors that is shown. And definitely at this point, the researchers got to know that based on the antibiotic effect, there is increase in the evolution of CRISPR-Cas immunity in the presence of bacteriostatic, whereas there is only minority of bacteria evolved CRISPR-Cas immunity in the presence of bactericidal. And they inferred that the fact that all the bacteriostatic antibiotics have different modes of action, they suggested that bacteriostatic antibiotics can promote the evolution of CRISPR immunity because they limit bacterial growth rates. And this is the second main part of the first experiment, that is to better understand the relationship between the bacterial growth and the evolution of CRISPR immunity using antibiotics. They calculated the OD values using the OD reader, and exponential growth rate in LB was determined by R using a software package growth rates. So again, they have taken the overnight cultures, performed dilutions, and then transferred into a fresh medium, and then they got the values using this optical density plate reader at 600 nanometer. So that's how they got to know that. And here you can see that effect of CRISPR-Cas resistance proportion. So how did they get to know the cultures were resistant to CRISPR and the effect of exponential growth rate on that? So their observation was chloramphenicol and tetracycline cause particularly slow growth, and this is associated with a large increase in the evolution of CRISPR immunity. But the interesting thing is ERM is exceptional because they found a correlation between exponential growth rate and the evolution of CRISPR immunity. So they always, when we do biological experiments, we should always do or give a proof to the audience that how did they found that ERM is exceptional. So they did Pearson's correlation, and when they considered all antibiotics, they got a certain value, but when ERM was included, they got a certain value. So this proved that ERM is a macrolide that mostly affect bacterial growth and gene expression at high cell densities. And it's just for our understanding that correlation is the perfect negative and positive relation, that is -1 less than the R, that is the Pearson's value, less than +1. And it infers that thus in the presence of all bacteriostatic antibiotics, most phage infection events will happen in cells experiencing reduced growth rates.
Bacteriostatic antibiotics promote spacer acquisition. So this is the second main part of our article. What is spacer acquisition? We are going to have what's called spacer DNA. Now, what's interesting about the spacer DNA is that it's not identical. Each segment of spacer DNA is going to be unique, and this puzzled scientists when they identified this back in 80s and 90s. But in the 2000s, what they found that spacer DNA, that the important DNA matches up perfectly with viral, especially bacteriophages DNA. And spacers are essentially history of old infections, so we won't be infected again. This is exactly the way your immune system works. On a much larger level, you are making antibodies. And then you have WBC that will envelop the invader. Effect of antibiotics on short-term growth of PF14 phage resistant clones. This is spacer acquisition assay. So here scientists have done an experiment and they have taken overnight culture and they have diluted it twice and they have incubated it. They have took a fresh medium and they have incubated it for 37 degrees centigrade for 30 minutes. And the incubated medium was shifted into 24 well microplates, and bacteriophages were added, and the name of the bacteriophage was DMS3 VIR, and again they have incubated it for 37 degrees centigrade for 3 hours, and the result was density of phage sensitive cells was measured by plating on the LP agar up to serial dilution in growth media, and density of Phage-sensitive cells was measured by directly plating on LB agar without serial dilution. This density was sufficient to prevent growth of sensitive colonies. So the smooth colonies which are resistant to both the phages and non-smooth colonies which are resistant to DMS3 VIR and sensitive to DMS3 VIR ACR IF1, and these are CRISPR immune. In each culture, the proportion of CRISPR-Cas immune clones within the total population of resistant clones was calculated. So this graph talks about a proportion of phage-resistant clones that acquired new spacers on the y-axis and antibiotics on the x-axis. So as the result, short-term infection experiments reveal that in the presence of all bacteriostatic antibiotics, more cells acquired CRISPR immunity, whereas bactericidal antibiotics had no effect. So this graph talks about this graph is the result of the first experiment. Across antibiotics, the frequency at which CRISPR immunity is acquired in these short-term experiments was significantly correlated to the levels of CRISPR immunity that evolved at 3 days post-infection. So this is the graph which talks about effect on CRISPR-Cas resistance and on the y-axis and effect on spacer acquisition on the x-axis. Yes. So here we can see two graphs which talk about there was no significant correlation between evolved proportion of CRISPR-Cas clones and CRISPR-Cas selection rate either in the absence of phage or in the presence of phage. So as a result, collectively, these data show that the increase in the evolution of CRISPR-Cas immunity in the presence of bacteriostatic antibiotics can be explained by an increase in the frequency of spacer acquisition.
And now let us move to the third main part of the research study, that is bacteriostatic antibiotics slow down progeny phage production. So the aim was bacteriostatic antibiotics may slow down phage development, providing a larger window of time for CRISPR-Cas immune system to acquire spaces from the phage prior to cell death or irreversible cell damage. And how they did was they performed one-step phage growth assay, and they performed it for 12-hour pre-exposure to antibiotics and 30 minutes pre-exposure to antibiotics. So they have again taken the 100-fold dilution with antibiotics, and after 12 hours, they added the phages. They vortexed it, incubated it. Again, they did dilution to stop the reinfection of the phages and also to limit the adsorption. And again, they vortexed. They added to the 24-well microplate. While the parallel processing was going on, the samples were taken at time which was equal to 0 and approximately every 20 minutes. And when they collected, after they collected, they have diluted in the M9 salt. Basically, M9 salt medium is the minimal medium for the bacterial cultures, and they plated on agar plate. So after this, they found out that all samples were chloroform treated and plated on PA14 lawn, and phage densities were measured. So while they were looking at the phage densities, they have found that bacteriostatic antibiotics did give them results, but bactericidal, they did not give any results because they were exposed to antibiotics for almost 12 hours, and the phage densities were very low to even measure that. So we should look at the 12-hour antibiotic pre-exposure for bacteriostatic antibiotics. So the black lines talk about the no antibiotic treated, and the red one talk about the antibiotic treated. So on the y-axis, we have the phage density, and x-axis the time. So initially there was increase in the phage density, but as we look on the right-hand side of the graph, there is decrease in the phage density for all the bacteriostatic antibiotics, that is trimethoprim, ERM, chloramphenicol, and tetracycline from top to bottom. So this tells that all bacteriostatic antibiotics caused a strong reduction in phage production compared with the cells cultured in the absence of antibiotics. And now this 30-minute pre-exposure to antibiotics, the only difference was in their experiment was their dilutions. So that they did it for 100-fold dilution, and this they only did for 25-fold dilution with antibiotics. And after 30 minutes, they added the phages, and rest all of these steps are same for the experiment. And they found the results for both bactericidal and bacteriostatic. The interesting results were for bactericidal antibiotics, but also bacteriostatics did prove their results. So the black ones are the no antibiotic treated, the red are the antibiotic treated. And except for ERM, as you can see on the screen, there was no delayed or impaired the formation of infectious progeny phages for ERM. Rest other all antibiotics, that is chloramphenicol, tetracycline, and trimethoprim, delayed or impaired the formation of infectious progeny phages. But the bactericidal antibiotics, that is ciprofloxacin, streptomycin, gentamicin, and carbamazepine, of course, ciprofloxacin and carbamazepine did not show any result. They were same as the control experiment, but streptomycin and gentamicin, they also delayed or impaired the formation of infectious progeny phages. And in our further slides, you'll get to know how the infection experiments actually worked in the presence of all these antibiotics. And finally, to understand whether some of these antibiotics might inhibit phage production, they performed highly replicated experiments, that is 96 bacterial culture per treatment of bacterial populations. They have taken in around 5 phage particles per infection experiment was done. So after 24 hours of infection, they measured how the proportion of successful phage amplification depends on the antibiotics present, and hence providing an estimate of their impact on the probability of successful phage infection, and that is what their aim was. And these are the results of the phage infection experiment. So the yellowish orange represents the failed infection experiments, whereas the blue one represents the successful results. And all the like trimethoprim, chloramphenicol, ciprofloxacin, and carbamazepine, in the presence of all of them, all infected populations ultimately produced new phages. And interestingly, there is a small but significant increase in the failed infections in the presence of erythromycin and tetracycline. While this may contribute to the evolution of CRISPR immunity, that is what they were discussing in the paper. And all the 3-star ones indicates the number of population with no phages. And we infer that with most infections being ultimately productive, the delayed production of infectious phage particles observed for all bacteriostatic antibiotics is thus due to delay in the phage eclipse period. So here the chloramphenicol, like they ultimately produced new phages. So in their further experiments, they have used chlorampphenicols at different concentrations to check whether even that is able to evolve CRISPR immunity in the cells. So we would like to conclude with this graphical abstract that in the presence of antibiotics, when bacteria are infected, in the presence of bacteriostatic antibiotics, to be more specific, there was slow phage development and there was increased in the spacer acquisition. But in the absence of antibiotics or in the presence of bactericidal antibiotics, there was rapid phage development as well as no spacer acquisition. So this is the main crux of the whole research paper. And a wide range, apart from the data that we presented, they were also, as I already talked about the chloramphenicol concentrations, they also showed the data related to these 3 experiments. That is, a wide range of chloramphenicol concentrations promote CRISPR immunity. Carbon sources associated with slow growth also promote the evolution of CRISPR immunity, the other environmental factors. And not only that, but also the clinically used antibiotics such as azithromycin and etc. They also used all such antibiotics which was involved in the CRISPR-Cas immunity in the bacteria. So if you are curious to know the results of the other 3 data sets in the research study, you can go and read the paper. And
future scope. CRISPR-Cas system can provide defense against a broad range of mobile genetic elements, including plasmids, which play a key role in transmission of antimicrobial resistance, AMR. Being able to trigger spacer acquisition for plasmids could therefore have important implications for constructing the spread of AMR genes. Identifying the ways to simultaneously promote spacer acquisition from plasmids and limiting the acquisition of spacer from phage could provide a powerful means to control pathogen abundance, virulence, and the resistance to antibiotics.
Thank you for your time and listening. Yeah, and now I would like to answer a few questions after the presentation, that like how this journey was when we were reading the whole article. I was really passionate about CRISPR technology since I was introduced since it was introduced to us, and thanks to Anil sir. And the minute we saw the article related to CRISPR in the list of articles, I just picked that. And the learnings were, I have learned new methods which I want to practically perform them, and to understand what experiment was it. And it was very essential to see how the scientists actually carried out their experiment, like spacer acquisition assay, one-step phage growth assay, the evolution experiments, and other experiments that were there in the paper, which really excited me. And I was understanding how the concepts were involved and how I can actually perform them when I am designing an experiment. So team building is like very important, and it actually gave me strength to make our presentation more effective. And as a team, as we are working in a team and discussing the work and connecting the different aspects in our learning would definitely help us. And the challenges that I faced was the difficulty of the paper. And as far as I have understood their work, to derive at the first hypothesis in the paper itself was the biggest challenge for me, because the results were not directly connected, but there was a lot of supplementary information that was required for me to understand in the paper. And most of the supplementary did help me to connect the dots. Thank you.
Are Manaswini and Gayatri going to share anything, or is that the end of the whole presentation?
Yes, sir. Yes, sir. Hello, everyone. Thank you for your time for our presentation. So, yeah, through Samyojana, the thing which I have learned is I have overcome nervous breakdown, because I have a lot of stage fear, and now I don't have it. And today I could able to give my presentation very well without stuttering and stucking at a point. And it has happened only because of Samyojana. And whereas if I talk about the article, the thing which fascinated me was spacer acquisition mainly. So that is the reason why I have took spacer acquisition as my topic. And so I don't want to go into further detail about it because I've already spoken about it. So, and talking about team coordination, I would say that Rachna is really someone to me who understood me very well and did all alone, because I and Manaswini had a lot of college presentations and college stuff to deal with, but Rachna completely understood, though being a third year student, but still she did it all alone. And she called us, I have done the presentation, PPT is done, we can have a mock, we can do that, we can do this if any changes, but still the presentation was perfect, but still she asked her opinion. So yeah, I would really thank her. And yeah, that's it.
Yeah. Yeah, bye, Salmot.
Was Manaswini saying something?
Yes.
Okay. Go ahead.
Okay. By Samyojana, I just mainly stick about time management, which I have that. Actually, I don't know how to manage my time, but due to Samyojana, I stick to my time management. That's the first thing I get it and learned it by Samyojana. And I really thank Gayatri and Rachna to support me in this Samyojana program. And thank you, sir.
Very nice. Thank you very much. And special kudos to Rachna for sticking it up, and I mean, like, being there for and a support for the first years. So I have to mention this, right? So there is definitely a majority of St. Anne's presence here. And I don't know if you are all aware that Sudha ma'am is actually a graduate of St. Anne's. So I hope you take the advantage and kind of bond with her, share some of your thoughts.
Know that.
And get to know and get her advice as well. So Sudha, do you want to go ahead, ask questions? I do have a couple of questions, but I can wait till you ask yours. You seem to be muted.
Sorry. Hi. Yeah, I am a St. Anne's alumni, and it's wonderful to see so many of you, thanks to Anil, being excited about science and scientific research. I was wondering, Rachna, can you share the presentation again?
Yeah, sure, ma'am.
Now, this is another thing. Don't stop sharing the presentation, because many a times once you're done with presentation, people would like to go back to some slides to ask specific questions. No? So that is one thing that you don't have to do till the host says, please stop sharing your presentation. That can be a cue for stopping it. Can you go out of the presentation mode? It might be easier that way.
Yes.
What software are you using to present? It's very interesting. I've never seen this before.
Ma'am, this is the PowerPoint. I didn't use any software.
I see, but somehow your thing showing up only in the center now.
They're using a phone to present
I see.
there.
Okay. Good thing. And these are all sort of a good job with the presentation. I learned some very new things. I had no clue about this. I think, who was it? Probably it was, let me try, Medha, who talked about how she likes spacer acquisition. Isn't that you, Medha, who said you like
Gayatri.
Ma'am, Gayatri, ma'am.
Oh, sorry, Gayatri. Thank you, Ma. Gayatri. Gayatri, so a quick question for you. I wasn't clear, maybe I missed it. How are they quantifying spacer acquisition in this particular paper? You see, they say proportion of phage-resistant clones that acquired new spacers, right? How are they quantifying this? Sorry.
Rachna, can you?
Yeah, ma'am, if I can take that question.
No, Rachna, just slide it.
Okay.
Hello, Rachna.
Yeah.
1 second. Actually, your screen, it's not visible properly.
I think she wants you to go to the full screen, full slide mode.
Yeah, now it's fine, Rachna.
Gayatri is trying to say something because we can't hear you. Have we lost Gayatri?
Is there?
Actually, so they didn't use any of the sequences and all, but based on the if they are resistant or if they were sensitive, so based on that they quantified the spacer acquisition in their experiments.
So okay, because okay. So they literally went by this plating technique and then just presence of the colonies, right? The other general question I have is, why do you have to dilute the cultures before you go on to do any of the analysis? Why is that pertinent and why is it important? If you go back to the slide, no?
Okay. This one.
Yeah.
So the general answer that I can give over here is maybe because if they have lawn of cultures, they can't actually see how the individual cells are reacting in their experiment. So in the dilutions, the cells being reduced and they can take those cultures for their experiment.
So it's literally about manageability and about being able to make robust observations. Is that why they dilute? Is that what you're saying?
Yes, ma'am.
Okay, and one final question would be about if you did not, if you were not this fascinated by CRISPR-Cas, you may please continue to be fascinated with it because it's a fascinating technique. What other technique would you sort of rely on to try and reduce AMR and things like that? Is there anything else, or is it, I mean, which might be equally good and which might circumvent some of the other issues that might come up with CRISPR-Cas?
CRISPR-Cas is one because I'm definitely passionate about it. So I can look into this field more than the other fields. But I guess I have to check out what are the other techniques on AMR. Like how can that help that?
Okay, in your reading, you didn't think about something else being probably equally exciting or equally possible more than anything else to be
No,
able
because
to overcome
CRISPR
it.
was only my focus.
But as a microbiology student, conceptually, can you think of other ways? You might not have specifics, but can you think conceptually what other ways you could address this problem?
Yeah, I mean, using less antibiotics can be one aspect to reduce antimicrobial resistance because we are used to take more and more antibiotics as time goes on. So that could be one
This is
answer
at
that
the
I
level.
can
This
give.
is at the level of human behavior. I'm mostly interested more at the level of the microorganism.
So in the article itself, they were talking about the surface modification resistance, which can also help in the antimicrobial resistant activity. So that could be the other aspect of it. So they can actually do the experiments related to that.
What do you mean surface modification? Sorry. Surface modification of what?
Surface modification of the bacterial cultures.
But that I thought was if you think about cause and effect, I thought that was more an effect, an outcome. Am I making sense?
So Rachna, my question might be relevant to that. So you kept saying, and I believe I asked you this question earlier as well. So they are talking about surface modification. I mean, it's almost like they invented or they use this term, but what is the basis for the surface modification? What is the biological principle behind the surface modification? What's happening to
Means,
the
yeah, so the bacterial cells are not adhering on the surface of their cultures, and that is why they were resistant to it.
How? Why? What is the biological principle there? So that's good. So it's about phage adhesion to or phage attachment to bacteria. But at the molecular level, what's happening? Why are bacteriophages not able to attach to the bacterial
Yeah, because
surface?
they have used one of the phage strain, that is DMS3 VIR ACR-IF1. So
We don't know what that is. What does ACR-IF1 mean? I have no idea.
so it is the anti-CRISPR a phage clone that they have used in the experiment, and when the Cas9 goes and binds to the sequence, there is like no effect, and the bacteria, the phage cannot bind on the surface, and that is how they got to know the surface resistance.
But I thought surface modification, it has got something to do with the pillars, right? The phage, what is it called? The pillars or pilus or whatever you call it. Isn't there a structural thing there which the virus has to bind to?
Yes, that is there. Okay, maybe I can check into that and get back to your question.
Yeah, I guess the take-home message is as much as CRISPR-Cas9 is extremely interesting as a technique, one thing to think about is the downsides of overuse of this technique. And two, if there can be a complementary technique that can also be used in order to address this problem in a more holistic manner.
Yes, ma'am, definitely.
Any other questions from the audience? Comments? Any advice, Sudha, in terms of?
No. I guess sorry, hang on 1 second.
Oh, more questions.
This, no, don't move. Just roll down, aim ke 2 slides ke niche. Yeah, very slowly. Yeah, very slow. Stop. I mean, go down, please. The plots, 12-hour antibiotic plots. Yeah. One thing I would have wanted you people to comment up front was about why the y-axis looks so different in both the situations.
Yes, ma'am. Yeah, so
bacteria,
actually
right?
the left-hand side graph was from the actual research paper, and the right-hand side was from the supplementary data. And they didn't put the y-axis like a lot, like where the phage densities are actually decreasing. They didn't put that in the actual result, but they were describing like as time goes on how they put that and how the phage density was decreasing
See,
more and
overall,
more.
right now, both of your x-axis are exactly the same. You're looking at a 3R profile. What
Yes.
is resulting in the y-axis to differ by an order of magnitude in the two sets of growth curves?
See, the y-axis on the left side is linear, but on the right side is exponential. The question is why? What have they done different?
Because that will have a very important bearing on the result that they're seeing actually in the first place.
Right. So
something to look into.
Yep. Yeah.
So I think one obvious answer or one obvious take-home message is look at your axes properly, right?
It's very, I think, very true. And that came up even in Shrishti and Sarah's presentation. I think one needs to be very observant about what the authors are trying to communicate in any graph or plot that they have in their papers. And especially if it looks like those double axes plots, right? It's very important to see what are the numbers on them and what do they represent. And many a times I've noticed that in papers, they will sort of compare across two sets where the axes are vastly different. And they won't even comment about why the axes are vastly different. And that I think is not cool. It's something that should be mentioned in the legend itself, figure legend itself. And if I were the author, I would make sure that I will make a one-line statement about it, or I will discuss it a little more detail in the results section. So this is something that I'll be careful about. Yeah, because also in this particular case, I can't help but think that this phage density in itself also has implications for why the authors see what they actually see, what the underlying mechanism could actually be in terms of spacer acquisition business, right? So that might have some implications is what I think.
Yeah, I also think in terms of experimental detail, that the initial density, the culture volume / maybe the dilutions might be in play. There might be a what's the right word? Pre-treatment of antibiotic, and suddenly a surge, something kind of a pulse and a chase kind of thing might be there, and for a particular reason. So I also missed this, but yeah, something we should look into in detail.
But they did mention in the figure legend that they have cropped the y-axis.
On which one?
On the left.
Right side or the left side?
The left one. The left is the main figure, so they have cropped that. They cropped the y-axis, and they have shown that cropped image in the supplementary information on the right-hand side.
It still doesn't explain.
Hey, sir, I want to make a comment on this figure. Like, if we look at the scale of y-axis, that also differs. Like here, they are taking from 10 to 1000, and while in the A figure, it's from 0 to 150. So when we look at the graph, it is kind of the squid one.
So you are saying that okay. So it is actually the same thing, but when you take it in an exponential phase, you'll actually see a much larger one. But if you make it linear, you actually should see a sharp slope. Is that what you're saying?
Yes, sir.
It's essentially the same thing, but when you change the
Exactly.
y-axis, it looks slightly different. Okay.
Yes, sir. So like the scale is what I see a different point. Like here they are taking from 0 to 150, while in the second figure they are graph they are taking the values from 10 to 1000.
So there's
So
essentially the slope difference. That's all. The way.
yes, sir.
Okay. So I think Rachna, you also it would have been better or nicer if you actually talked about the anti-CRISPR, the variants, right? What is the difference between PA14 and PA14 IF or 1F and the corresponding phages? That would actually, at least to me, it would have given me a little more clarity about the assays, the expected results, what's happening and things like that. Because the beauty of this experiment is in the strains, the use of the different strains and their properties. And I think I know that you practiced a lot, and I know I pushed you a lot about time, but you could have slowed down a little bit and you would have been fine.
Okay. I'll make a note of those things, and I'll definitely check out the bacterial strains and the phage strains and their effects. And I'll get back to you with that.
Thank you. So Nashra says, ask us
Two
so
comments
yes,
to
no,
have.
please go ahead. Yeah.
This is talk 2, meningioma group.
Yes. Okay.
You're muted.
Can I stop presenting now?
Yeah, you should. Yeah, thank you.
Yeah.
Thank you so much, sir and ma'am.
Thanks.
So the meningioma of Medha is here. Medha, you're the only one from that, right? Kiravani and Nidhi are. I can't see Nidhi. I can't see Kiravani.
Medha, would you have the presentation? If you don't, it's okay.
Ma'am, I don't have a presentation. My teammates, I think they went.
Oh, you're the one with the audio problem, man. You need to change your headset. Extremely difficult to hear you.
Yeah, ma'am, I'll do that.
Yeah, I think it has, my suspicion is it's only because of your headset. One question I don't know which one among the three of you talked about cDNA being a double-stranded DNA. I think the definition of a cDNA is slightly different. How you make the cDNA is something that is intrinsic to how you define the cDNA. Do you guys know what exactly a cDNA is?
No, ma'am, not me.
So typically information in an mRNA, when it's used to make the DNA, that particular DNA is called a complementary DNA. So when you have one such scenario, for the most part, you probably have a hybrid DNA, hybrid molecule, which is an RNA DNA hybrid. So it cannot be a double-stranded DNA to begin with. That cDNA then can be separated by techniques from the mRNA or the RNA in this experiment, which was not an mRNA. And then you can amplify it, and then you can get double-stranded versions of it, but not before the PCR amplification. So cDNA is not a double-stranded DNA, not to my knowledge at all. Yeah, that I had one comment about. And I don't know who talked about gene ontology. I wanted a little more details about what you mean by gene ontology.
Here, the gene ontology is they are trying to understand the tumors and how the gene replication do occur here. So
ontology means what? When you say ontology, what do you mean by that?
I think it's the study of how the gene variation occurs.
Okay, you should go look it up. But overall, when you are using some terminologies like this, no, it's okay to assume that I'm not a person who does most of the techniques that you talked about or deals with it on a day-to-day basis about the techniques that you talked about in talk 2. So what will be nice is to sort of preemptively communicate some of these things up front, so the speaker, so the audience is not lost when the speaker says something. And then after that, I'm constantly thinking about what did she mean by this? Yeah, what does that mean? And stuff like that. And before I know, I'm not concentrating on what you're saying subsequently. And I'm sitting and pondering about this. So what might really help, or what will really help, is for you to assume that the audience is a layperson might not know much about whatever you're going to be talking about. And it's okay to take extra couple of minutes to explain things. And anytime you put any data set, and this holds good for all the three groups, anytime you put any data set on a slide, you need to explain it in full. You don't have to explain all of them. If all of them have common axes and if it's a common assay, you take a second to explain one of them, then the rest of them become very self-explanatory. But if you won't do that, then again, the audience is lost in trying to understand the plots. And before you know, what you're saying becomes a little blurred because we are sitting and concentrating on these plots to understand them better, no? Or on this data set to understand it better. This is a general take home something for all of you to remember. And this is something I learned with time, that if you cannot communicate what was meant to be communicated in a given experimental data set, it's going to be very hard for you to keep the interest of the audience. Very quickly, it'll be kind of, they'll be lost in their thoughts. And these days, there are a lot of other distractions too. So someday, I hope, though, this happens in person. You started off and then it was going on online only, right, Anil?
And there was a small chance that this would have happened in person, but we could not pull it off in a short notice. But going forward, that's the plan.
Yeah, because speaking in person is like world apart. You are physically in the same room as your audience. You have to engage with them. You need to make eye contact with all of them, or as many as possible. You will never be able to see an individual, but you'll see sections of individuals. So that, I'm sincerely hoping for you people to have that experience too. It might be a little difficult to coordinate across India, but maybe we can think of Samyojana in regions and then have centers or institutes be able to host it and have more students come and do this. And this is something I'll talk to you.
You're stealing away all my, basically exactly the thoughts I've been working on. Yeah.
Yeah, wonderful. I'll be happy to brainstorm with you more, or in any way I can.
Yeah, I was going to come to you with those ideas and to actually tease them apart, find out what the best format could be. But yeah, we'll talk more about that.
Sure, absolutely.
Thank you so much for being here for this session.
No, my pleasure. My pleasure. And thank you so much for having me. It was fun.
Awesome. Thanks again. So we'll close this session, go get a quick bite, come back. We have planned to meet at 2:30. So that's a decent time, I think, for having a quick lunch. Sudha, I don't...
I will have to excuse myself. I have to go and meet a friend who had a surgery. So
that's, yeah. So absolutely fine. Again, I know that being a Sunday, being remote, it's very difficult to dedicate the whole day. It would be very different if it's an in-person thing. You are stuck in a nice way.
Yeah, you're stuck in a happy way. And then I think that makes a world of difference for the kind of interactions that are possible and for the learning experiences. Yeah, exactly.
Again, thanks again to everybody. So we will meet in about 35-40 minutes to resume the last session on neurobiology. And Sudha, just in case if there are students, I'll send you the link and they can join in if they're interested. Any students around, if you happen to see anybody or if you think of anyone, if you want to pass it along, I will send you the message. You can pass it along, so that if they want to attend and listen to some the student presentations.
You're talking about me. Say that again.
In case you know of students who are interested in neurobiology at ISEP.
Absolutely. Yeah. So I don't know how you communicated about this event to different. I have a whole bunch of questions, because there would be students even from ISEP Pune who would be very happy to participate. We
were doing a low key. We were testing ourselves whether this model is going to work. So we deliberately kept it a low key. But yeah, right now
I still think we should target student populations where the chances of doing this is not very prominent, or not very, it doesn't happen on a regular basis. For example, students in ISEP Pune might have more of an opportunity to do this because of the way we structured our courses, though COVID has played havoc with all of that. But in this, I still think it is a good idea to think about a few levels of how we want to do this. And I can't wait to see this become a big thing in the subsequent years. There'll be a lot of
Absolutely.
takers, is my suspicion.
I'm looking forward to that.
Sure, sure. Bye, Anil. Thank you so much again for the time.
Have a nice
Thanks,
day. Take
Peter.
care.
Thanks. Bye, 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 bye,
Bye.
bye. Ciao, ciao.
So what I'm going to do now is just pause everything. Let's go take a break, get lunch, and meet at 2:30. Does that sound good?
Absolutely, sir.
Yes,
Yeah,
sir.
and
Yes, sir.
we haven't done what we typically do, which is okay because we have the video. But should we do? I mean, I know you guys, some of you are interested. Should we do the what's it called? Everybody show their face and have a photograph, if it's possible. But otherwise,
You'll
it's
have
fine.
to give us some time for that.
Some time. Sounds good.
Yes.
Okay, let's meet at 2:30.
We can do that.
Yeah. Bye.
Bye.
So let's meet in half an hour.
So is everyone leaving? I just wanted to know if we can hang around for a second. I mean,
So
not.
what I'm going to do is this. So I'm going to stop. So right now the session is going is live. So I'm going to end this session, right? End as in like I'll stop